morning, everyone. Good morning, everyone, and welcome to the symposium on recent trends in pharmaceuticals organized by Department of Pharmaceutics to Vidyanathan College of Pharmacy. It is my great honor to welcome our chief guest, Professor K.P. Chandrasekhar Garu, Vice Chancellor, Krishna University, Andhra Pradesh. Sir, it is our privilege to have you in this symposium. It is my pleasure to invite the speaker of the day, Professor K.P.R. Chowdary Garu, Academic and Research Advisor, KVSR.
Viveraka, you have muted yourself again.
Is it audible, sir, now?
Hello? Hello?
Yeah, audible. Go ahead.
Okay, it's all.
Yeah.
Okay,
It is my pleasure to invite the speaker of the day, K.P.R. Chowdary Garu, Academic and Research Advisor, KVSR, Siddhartha College of Pharmacy, Vijayawada, and Research Director, Vikas Institute of Pharmaceutical Sciences, Rajahmundry. I would like to welcome Professor L. Venugopal Reddy Garu, Advisor cum Director, SVET, and Director Sudarshan Garu, Director Q&D, SVET, to the 3 days e-Symposium. Thank you very much, sir, for gracing the occasion. I wholeheartedly welcome our beloved.
Hello?
Madam.
Principal sir, you can take over now. Already
Yes,
the formal introduction is done. You can take over, please.
Yes, sir. Sir, good morning to all of you. I will be giving the welcome address. Good morning, eminent scholars and honored guests. While it is obvious to feel happy to deliver the welcome address, I am also excited and feel blessed about this role. This 3 days e-Symposium is being conducted with the intent of active and interactive learning, organized by the Department of Pharmaceutics, Sri Vidyanathan College of Pharmacy, Tirupati. I am extremely thankful to the management of Sri Vidyanathan Educational Trust for giving me this opportunity to serve this great institution and always also blessing us with all the necessary infrastructure, permissions and support for the conduct of this 3 days National e-Symposium. I happily welcome Professor L. Venugopal Reddy Garu, Advisor cum Director of Sri Vidyanathan Educational Trust, former Chairman of AP State Council of Higher Education, former Vice Chancellor of Andhra University and Nagarjuna University, and also retired Professor of Sri Krishnadeva University, Anantapur. I am extremely happy to welcome today's keynote speaker and chief guest, Dr. K.P. Chandrasekhar Garu, Vice Chancellor, Krishna University. He is an eminent educationist and scientist. His idea of student centricity has ushered in several developments towards academic achievements, student employability, entrepreneurial achievements, amongst many other noteworthy laurels to be mentioned here.
Hello, sir.
I wish to extend a warm welcome to Dr. K.P.R. Chowdary Garu,
ex-principal, Department of Pharmacy, minimize chesi petna,
Andhra University, and Academic and Research Director, KVSR Siddhartha College of Pharmacy, Vijayawada, and Research Director, Vikas Institute of Pharmaceutical Sciences, Rajahmundry. He is an ardent orator and a devoted teacher who has mesmerized scholars and people of varied caliber with his intellect and charm all over the country. A noble endeavor of knowledge summit such as this needs the support and blessings of senior functionary of the trust. The College of Pharmacy deems it to be its privilege to be supported by our beloved Director Quality and Development, Dr. I. Sudarshan Kumar Garu. A hearty welcome to you, sir. I am extremely happy to welcome Dr. S. Mohan Lakshmi Garu, Vice Principal, HODs, professors, program coordinators, faculty members, students and esteemed audience who have registered to this webinar and are watching this webinar live streaming of this program from diverse places from across the country, from nooks and corners. May I also state that we have the privilege of receiving a total of about 700 registrations so far for this program.
Oh, wonderful.
I hope that all of you will have 3 most productive days of interesting and stimulating sessions. I once again extend a warm welcome to everybody present here. Thank you very much. Thank you very much. Over to you, Eureka Madam.
Virake, madam, unmute yourself.
Thank you for delivering the welcome address, sir. Now I call upon Professor L. Venugopal Reddy Garu, Advisor cum Director, SVET, for his valuable presidential address.
Yeah. Once again, good morning to all of you.
Morning, sir.
Good morning.
I welcome all of you to this wonderful symposium organized by our Department of Pharmaceutical Sciences, Sri Vidyaniketan. And of course, Professor K.P.R. Chowdary was my previous colleague. When I was Vice Chancellor of Andhra University, he was my colleague, and of course, he helped me in several respects. And also, of course, others are also known to me either directly or indirectly. I once again congratulate the Department of Pharmaceutical Sciences for organizing this wonderful symposium at this point of time. When everybody speaks about COVID and the vaccine and all that, I think this is the appropriate time to organize this kind of a symposium. So of course, the topic given for this symposium is recent trends in pharmaceutical research. And of course, as we all know that, the pharma industry's lifeblood is research and development. Every pharmaceutical industry need to spend more on research and development. Otherwise, their survival itself is questionable. So on an average, 17% of their revenues are spent on research and development. Of course, the recent trends in pharmaceutical research include adoption of artificial intelligence by pharma industry and new antibiotics discovery and personalized medicine. And of course, there are several things that what are happening in the recently as far as the pharmaceutical research is concerned. And we are all now concerned. Every common man is also now talking about vaccine. Everybody is waiting, and everybody speaks about which country, and there is a cutthroat competition within the country as well as outside the country as far as getting this vaccine for COVID-19. So the future of pharma depends on embracing technology. It's about developing biological drugs as well as vaccines. So I think this is enough. And I really congratulate once again the Department of Pharmaceutical Sciences for organizing this symposium. I wish good luck, and I'm very happy to learn that there are around 700 people who have registered for this symposium. That itself is a really great achievement. So I congratulate once again, and I wish this program for another 3 days will go very successfully. Thank you. Thank you for giving me this opportunity.
Thank you very much, sir. Thank you.
Thank you very much, sir, for sharing your thoughts and blessing this symposium. It's a time for director's note. Now I request our beloved director, Dr. I. Sudarshan Kumar Garu, to address the gathering.
Distinguished guests of the inaugural session, my mentor and my senior professor, Professor L. Venugopal Reddy Garu, who is the advisor cum director, Sri Vidyaniketan Educational Trust. At the same time, the chief guest of the inaugural function, Professor K.V. Chandrasekhar Garu, who is the Vice Chancellor of Krishna University. At the same time, today's guest speaker, Professor K.P.R. Chowdary Garu, who is advisor, academics and research at KVSR Siddhartha College of Pharmacy, as well as the advisor at Vikas Institution, and all my esteemed colleagues, participants of this function, of this inaugural session. I'm extremely delighted to be associated with this event. Firstly, it is a very fantastic transition that education has really taken up, because what is supposed to happen after 5 to 6 years is now happening today. The transition of this education into online education, hybrid mode of education has been taken up. And today we are all sitting at our homes as well as in different places. And at the same time, we are able to really meet and discuss about one of the frontier areas of research. That is what is happening. Today, we should really be happy for taking this smooth transition at a very early stage and getting into the online education. Now, firstly, let me just, on behalf of Sri Vidyaniketan Educational Trust, let me thank our Honorable Chairman, Dr. M. Mohan Babu Garu, who has provided the fantastic institutions really to work with and produce some of the best alumni that are possible from these institutions. Nextly, let me take this opportunity again on behalf of our Honorable CEO, who is again a very proactive leader who is working with all of us and taking these institutions to the next level.
Good.
We have a great team of people who are working with us in this group of institutions like Professor Venugopal Reddy Garu. There are umpteen number of teachers like Professor Balaji, Professor Mohan Lakshmi. We are doing fantastic research at this group of institutions, and very close to 2.5 crores worth of funding is there, and we have very good interaction with different funding agencies as well as with the different industries. Now, Professor Venugopal Reddy sir was talking about the need for taking up advanced research. With the advent of technology, with the intervention of novel technologies that is happening, really it is becoming very easy for the people who are working in different areas like medicine, maybe pharmacy, that is really taking up a faster pace. Today we see there are close to 100 different types of vaccines at different parts of the world being taken to testing level. That itself is a testimony of intervention of technology and the amount of research that is happening in the area of pharmacy as well as drug and pharmaceutical research. Now, the other area which is very important where a pharmaceutical person can really work is a electronic intervention in terms of device management, where the concept of sensors can be brought into testing, because the very concept of addressing a pandemic or an epidemic comes into way of testing, tracing, as well as treating. The very concept of testing with respect to taking up device-related technologies will be one more area where a pharmaceutical person with little bit amount of technology, with little bit of electronics can really look into. For young researchers, I just would like to tell you that normally in a very fundamental way of discussing about 3 I's. When you are a student, you should be really good in imitation, because while doing the practicals, you should be in a position to really do the protocols. You should be in a position to do the good experiments that are there in the list of experiments in a proper way. That itself is, you need to imitate and then get expertise, skill development that should be there. When you go to the next level, when you take up serious research, the concept of innovation comes, where you little bit of tweaking into the process, where from imitation level, you just get into new knowledge, new technologies, new things that you will be positioned. Once you keep on pursuing this concept of innovation and you start publishing, you start patenting, you should start technology transfer, the next level comes into picture, where at some point of time, with the level of interest you have, and if you are very lucky enough, you'll get into the third stage, that is invention. Invention is a rarity among academic community, but still the third level, that on persistent efforts, you'll be getting into that. So young students and young researchers, I just like to advise that it is always better to follow these 3 I's, follow the imitation model, because once we read a paper, it's not just reading and getting the knowledge out of it, really doing that same amount, imitating that experiment in our laboratory, whether it is feasible, whether the results that are given are possible or not, will help us to really know what is happening. Next level is like innovation, and then final level to the invention. So it is really, it's a really great stage of research where we take up from this level to the level of invention. So I'm really happy that the College of Pharmacy has been associated with the experts in the field, Professor K.B. Chandrasekhar Garu, at the same time Professor Chowdary Garu, who is being part of this event. And I hope that the participants as well as the faculty, who are all student as well as faculty participants, will be benefiting out of this a large way, and take up a good amount of research that will be not only contributing their own personal glory, and at the same time they'll be for their institution glory, and finally they'll be contributing to the economy and research development of the nation, that is India. Jai Hind, and I wish everybody a happy and healthy stay at your places. Thank you very much. Thank you for giving this opportunity.
Thank you, sir. Thank you very much, sir.
Thank you very much, sir, for addressing the gathering.
Now I request Dr. S. Mohan Lakshmi ma'am, our Vice Principal, to introduce today's chief guest.
Madam, unmute yourself.
Yeah, hi everyone. Very good morning to all of you. I hope you all are safe and healthy wherever you are during this calamitous situation. I extend a warm welcome to each one of you to the journey of this 3 days National e-Symposium. Today we are very delighted and honored to have with us an eminent and prominent personality, the Vice Chancellor of Krishna University, Machilipatnam, Professor K.B. Chandrasekhar Garu, as chief guest for this National e-Symposium. I have been given a pleasant task to introduce our chief guest. I am happy and honored to do that. Professor K.B. Chandrasekhar Garu was born in the Anantapur district of Andhra Pradesh. He has graduated in science from Sri Venkateshwara University, Tirupati, and he has earned his post-graduation in chemistry from Sri Krishnadevaraya University, Anantapur. He received his MPhil degree for his research on the topic Studies on Mixed Ligand Complexes of Biological Importance, and he received the doctorate for his research on studies in the chemistry of metal pollutants from Sri Krishnadevaraya University, Anantapur. Professor K.B. Chandrasekhar Garu joined as assistant professor in chemistry at JNTU, Hyderabad in the year 1991 and joined as associate professor in the year 2000 at JNTU, Anantapur, and then elevated to professor in the year 2008. He has served in the various administrative positions such as head department of chemistry, controller of examination, director of evaluation, director research and development, coordinator for Inflibinet at JNTU, Anantapur. He also served as director Oil Technology Research Institute and director Foreign Affairs at JNTU, Anantapur. During his tenure as head, he was instrumental in implementing many quality practices in the department, such as successful implementation of TechIPS project, developing analytical research labs, modernization of existing laboratories, etc. He has played a pivotal role in the revision of syllabi for autonomous institutions as Chairman Board of Studies of JNTU, Anantapur. He played an active role in curriculum designing as Chairman and Member Board of Studies in the Department of Chemistry, Pharmacy, Biotechnology and Sugar Technology in both JNTU Anantapur and JNTU Hyderabad, and also to many of its constituent and affiliated institutions. He served as university nominee for the governing body of many affiliated institutions under JNTU A jurisdiction. The leadership provided by Dr. Chandrasekhar Garu as director for various portfolios at JNTU A has resulted in implementation of many quality practices such as introducing EDEP system in the examination, framing standard quality guidelines for the PhD students, introducing anti-plagiarism check software like Turnitin for PhD thesis, conducting research review meetings during the various stages of doctoral research to access their progress, uploading of PhD thesis in Shodhganga, and introducing pre-PhD classes for the research scholars. He is the recipient of the prestigious Best Teacher Award conferred by the Government of Andhra Pradesh in the year 2012 and honored with Research Excellence Award at Indo-Global Education Summit by the Indus Foundation in the year 2017. He is the recipient of Best Research Paper Award by India Drugs Association in the year 2005. He has organized many conferences and seminars by various funding agencies, sponsored by various funding agencies such as AICTE and UGC at JNTU Anantapur, and delivered lectures and research talks at reputed institutions across the globe. He has guided so far 73 PhD theses and guiding 8 PhD at present. For his credit, Dr. Chandrasekhar has 265 international and 127 national publications. His H index is 22, I-10 index is 54, and citation for his research article stands at 1,860. He has authored books on engineering chemistry, instrumental methods of pharmaceutical analysis, and also authored chemistry laboratory manual. He is a life member of many learned professional bodies and societies. He visited countries like China, Thailand, Dubai, Sri Lanka, Malaysia, Ethiopia, Indonesia, and Germany to present in reputed seminar and conferences and popularize his research findings. He has also actively involved in many social and rural outbound activities implemented by the government of Andhra Pradesh at JNTU Anantapur. We are delighted and honored to have you with us, sir, and I request you to formally inaugurate the symposium.
Vivereka, madam.
Thank you, ma'am. Now I call upon today's chief guest, Professor K.P. Chandrasekhar, sir, to inaugurate the session and deliver his inaugural address.
Sir, now open. Sir, kindly give the permission to open the.
Head. Head. Hello?
Sir, okay.
Okay.
So there, sir.
Open.
Namaste, sir. Sir, now inaugural talk.
Audible?
Yes, sir.
Okay. So first of all, my sincere thanks to the chairman of Sri Vidyaniketan Group of Colleges, Dr. M. Mohan Babu Garu, the CEO, M. Vishnu Garu, the principal of the college, the Professor Anne Balaji, the vice principal, the Mohan Lakshmi, the program coordinator, Professor Sharvan Kumar, Director Sudarshan Kumar, Venugopal Reddy Garu, and the eminent personality in pharmaceutical sciences, the Professor the K.P.R. Chowdary Garu, the other audience. So good morning to one and all. So today, the Sri Vidyaniketan College conducting the 3-day national e-Symposium on the recent trends in pharmaceutical research. So in this topic, the last decade has seen the rapid technology, the progress, but with increasing demands for the safe and efficacious drugs. The technological solution that reduce the barriers to clinical success will be of more interest than those that simply shift the bottlenecks and necessitate the new compromises in the discovery and development process. India's growing economy, coupled with its changing epidemiological profile with the cardiovascular problems and other chronic diseases that make it a strong candidate to become a future powerhouse of the research and development and manufacturing in pharmaceuticals. In addition, the clean water, the rising incomes, and better health infrastructure for the nation are contributing to an aging population with a greater demand for different types of the pharmaceutical drugs. Some of the leading domestic pharma manufacturers are emerging as innovators, both in-house as well as through external collaborations. patients are also the beginning to benefit from research advancement in the Indian industry. Nowadays, the pharmacy research is going on, the collaboration with the fields like the pharmaceutical sciences, chemistry, and biotechnology and engineering to develop the vaccine, diagnostic, and therapeutic. The pharmaceutical research involved in following areas like to develop a new chemical entity, to formulate a new combination for better effect, to improve patient compliance, to use natural products for the treatment of the various diseases, etc. But the several more active areas of the research that added to the list. The adoption of artificial intelligence by pharma and biotech. With all the type around artificial intelligence, nowadays it is a hard to surprise anybody with this trend in pharmaceutical research. However, it should be noted that artificial intelligence driven, the companies really started getting traction with the big pharma and other leading life science players with lots of research partnership and collaborative programs. A potential of artificial intelligence based the tools is now explored at all stages of drug discovery and development from the research data mining and assisting in target identification and validation to helping come up with the novel lead compounds and drug candidates and predicting their properties and risks. And finally, artificial intelligence, the software is now able to assist in planning the chemical synthesis to obtain compounds of interest. The artificial intelligence is also applied to planning preclinical and clinical trials and analyzing biomedical and clinical data. Next one, expanding the chemical space for drug discovery exploration. The key element of hit exploration is the access to an expanded and chemically diverse space of drug like a molecule to choose the candidates from, especially for probing a novel target biology, given that existing compound collections at the hands of the pharma were built in part based on the small molecule designs targeting known biological targets. The new biological targets requires the new designs and the new ideas instead of recycling excessively the same chemistry. Next one is targeting RNA with small molecules. So this is a hot trend in the drug discovery space with a continuously growing excitement. Academics, biotech, startups, and the pharmaceutical companies are increasingly active about RNA targeting, although uncertainty is also high. The next one is new antibiotic discovery, phenotypic screening. A small molecule, the drugs had actually been discovered using the phenotypic screening than target-based approaches. The technological advancement in pharmaceuticals is bringing hope to the patients. We are improving the healthcare through the use of innovative the digital technologies. So, in the following section, let's have a glimpse at some of the great technological advancement in the pharmaceutical field. The precision medicine, an approach that merges clinical and molecular information to understand the biological basis of disease. M-Health sensors have been developed with several M-Health apps for clinical research on iPhones. The apps are targeting the diseases such as diabetic, asthma, cardiovascular disease, and breast cancer. The 3D printing builds the pill by spreading the layers of the drug on the top of one another until it reaches to the right dose. It is porous enough to dissolve quickly. It is a beneficial for patients who have difficulty swallowing their medication. The nanotechnology process that helps in the delivery of anti-cancer drugs and to reduce toxicity. The nanotechnology can track the patients that take their medication to encourage the increased adherence. The nanobots are made up of the two different words, that is nanotechnology and robots. It can be consumed by the patient in form of pills or injections. The synthetic 3D, the tumors provide a better look at how the tumor behave and a more accurate measure of how they respond to treatment. A chip that is capable of stimulating tumors, the micro environment, and plan to use the new system to test the effectiveness of the nanoparticles and drug that target cancer. Nowadays, the pharmaceutical research centers are introducing the new and advanced instruments which helps in healthcare and final treatment for the various diseases. The microcapsules and nano coating for delivering the drug to specific site of the body. The drug experimentation on simulation models. Earlier, the drug experimentation is done on animal or human beings, but now the research institutes are doing research on simulation model drug testing. The drugs will be tested on simulating model physiology of the human body with the use of supercomputers. By using a simulation model, the testing, no one would get hurt in the drug testing process, etc. So, thank you very much for giving this opportunity, and I wish all the best for future. Thank you, one and all.
Thank you, sir.
Thank you very much, sir, for your valuable note. Now it's time for the virtual felicitation to our chief guest by convener Dr. Anna Balaji Garu.
Sir, we are very happy to receive you. We are obliged that you have accepted our invitation in the middle of your busy schedule. Please kindly accept this token of appreciation e-certificate, which will be emailed to you. A hard copy will be posted to you once the college reopens. Sir. Once again, thank you very much, sir, for accepting our invitation and joining this program. Thank you.
Thank you very much, sir. Chandrasekhar sir, thank you very much.
You're always welcome.
Thank you, sir.
Yes, sir. Thank you.
Very nice words, sir. You have covered all the areas, what are happening now in terms of electronics, artificial intelligence. Thank you very much for your useful words and insights.
Sri Vidyaniketri is my college.
Thank you so much.
Thank you, sir. Thank you, sir. Thank you, sir.
Welcome.
Thank you all, sir. I call Mrs. A. Sweta ma'am to read the profile of today's keynote speaker.
Good morning, respected dignitaries, faculty members, and my dear students. It gives me a great pleasure to introduce today's guest speaker, Professor K.P.R. Chowdary Garu. Professor K.P.R. Chowdary Garu is one of the senior most professors of pharmacy in India, having 44 years of experience in teaching, research, and 10 years of administrative experience as University HOD and principal. He has served as lecturer, reader, professor, HOD, and principal of AU College of Pharmaceutical Sciences, Andhra University during 1977 to 2010. He was the founder principal of KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada during 1994 to 2000. He was also a chairman for Board of Studies in Pharmacy in JNTUK, Kakinada during 2010 to 2017. Currently, he is the Research Director of Vikas Institute of Pharmaceutical Sciences, Rajahmundry, since 2007, and as an advisor for KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada since 2017. He was the member of various committees like AICTE, NBA, and NAC, and also as a Pharmacy Council of India inspector since 1980. He was the chairman and member of Board of Studies in Pharmacy for several universities. He was the co-convener and member of Scientific Services Committee of Indian Pharmaceutical Congress Association during last 10 years and elected as a national convener of Indian Pharmaceutical Congress Association for 3 years, that is from 2019 to 2021. He was the member of B.Pharm Syllabus Committee by Pharmacy Council of India, New Delhi for the academic year 2017 to 2018. He is having 9 years of industrial experience as a research consultant and research advisor. He has published 446 research publications with 3022 total citations of all the publications. He has published 2 books. He has presented more than 72 research papers at various conferences and seminars. Professor K.P.R. Chowdary Garu has delivered nearly 70 lectures as a chairman and resource person at various conferences and seminars. He has carried out 4 research projects and having 5 patents. He has guided nearly 120 M.Pharm and M.Phil projects and guided 100 PhDs. He was awarded 12 national and 6 state level awards. He has received various prestigious awards like Eminent Pharmacist Award in the year 2019, Lifetime Achievement Award in Pharmacy in the year 2017, Academic Excellence Award in the year 2015, Professor C.J. Sishu Award in the year 2004, Indian Pharmaceutical Association Fellowship Award for the year 2007 and 2009. Principal of the Year Award for 2008 and Pharmacy Teacher of the Year Award in 2003. He received Best Paper Award prize from Indian Pharmaceutical Association. He has also received Professor M.L. Khorana Memorial Award for Best Research Paper in 2001 and 2004. As the President of Indian Pharmaceutical Association for Rajahmundry Local Branch, he has received Best Local Branch Award for 4 times. He has received state awards like Best Teacher Award for the year 2007, Dr. Sarvepalli Radhakrishna Award for Best Academician of the year 2006. He received Eminent Pharmacy Graduate Award in the year 2006. He has received Eminent Pharmacist Award in the year 2004. He has also received Andhra Pradesh Scientist Award in the year 2002 and Best Researcher Award in the year 2000. It is my privilege to introduce such an eminent personality, Professor K.P.R. Chowdary Garu, to today's session. Thank you for giving me this opportunity, sir. Thank you. Over to Divireka, ma'am.
Thank you, ma'am. Now, without further ado, we will turn the session over to Professor K.P.R. Chowdary Garu to deliver his lecture. Sir, you can please start the presentation, sir.
Sir, can you share the PPT, sir? Your PPT. Sir, unmute yourself, sir.
Tarun sir, phone చేయండి.
Okay, sir. Now, screen share, the PPT is already shared. In the beginning, I kept. Okay, wait. Here, this is one. Okay. Okay, sir. Okay, is it clear, sir?
Yes, sir. Yes, sir.
Okay. Good morning, all. Respected Honorable L. Venugopal Reddy Garu, former Vice Chancellor at Andhra University, where I worked for nearly 35 years. And also he served as Vice Chancellor at Acharya Nagarjuna University. That time also I have a good rapport with the Department of Chemistry and Biotechnology of ANU. Several times I visited and met Professor Venugopal Reddy Garu. With his blessings only that we could do some sort of effective research in Andhra University. He always encouraged the researchers to do the best, and with his blessings only, we, the pharmacy department at Andhra University, could deliver some effective research in pharmacy. And the other dignitary today, K.B. Chandrasekhar Garu, and now he is, as you know, that he is the Vice Chancellor of Krishna University, Machilipatnam. Our Siddhartha College of Pharmacy is affiliated to Krishna University only. And earlier, when he is in JNTU Anantapur, and he used to look after the pharmacy also, along with the chemistry. And that time, I also met him several times, particularly for the evaluation of the PhD thesis and conducting evaluations, etc. And so I consider it as my fortune today to deliver my presentation in the presence of two great people, L. Venugopal Reddy Garu and K.B. Chandrasekhar Garu. Good morning, both of you, sir. And then the beloved principal, Balaji, Dr. Balaji, and the Sudhakar Kumar, CEO, and the convener of this program, Dr. Sarvan Kumar, and other learned faculty, and learned faculty members registered for this program, and prosperous students. Good morning to all of you. And first of all, I thank the management and the principal and Dr. Sarvan Kumar particularly for giving me this opportunity to interact with all of you on through this e-platform. And all of you know that we are suffering with the COVID-19 pandemic disorders. We cannot move out, and that made a revolutionary change in the educational system, probably, because earlier in pharmacy particularly, we don't have any of these online seminars and programs, except now and then we used to have an online lectures by eminent people abroad who cannot come out to India. Is rare, but today it has become so common, and it's a change, really good change. You can interact with any person anywhere in the world, and for the benefit of the students and faculty. So I congratulate the organizers for taking up this symposium, 3 days program, particularly concentrating on the research area. So current trends in research in pharmacy, and already our guest Venugopal Reddy Garu as well as the Chandrasekhar Garu has highlighted what is the importance of research. And all of you know that particularly research is vital for everyone. Without research, there cannot be any progress for the institution as well as individuals. But particularly in the pharma, the entire pharmaceutical industry is a research-based industry. And without research, they cannot survive, and hence the research is vital for pharmacy at all stages. That is the reason why those who are responsible for formulating the course curriculum for pharmacy courses introduce the concept of research at every stage in the B form. In the 8th semester, there is one full semester is earmarked for doing a project research work. In M.Pharm full 1 year is earmarked since long, not now. And PhD, of course, it is always everywhere by research. It is a research degree. And hence, the recognizing the importance of research in industry, our regulatory bodies also more concerned about introducing the concept of research in the pharmaceuticals. So also is the case at higher level in all other areas like in the chemistry, biotechnology. Okay, these pharmacy, chemistry, biotechnology, they are all one only. Because a good interaction between these three only lead to fruitful research. In isolation, we cannot do good research. So certainly, a collaborative efforts between the chemistry, biotechnology, and pharmacy leads to success. And you know that the essential purpose of research is only in pharmacy is to generate new drugs, new devices, new means, new treatment options, etc., for better use of healthcare, to provide better healthcare. So in that way, when you think about the research, so chemistry forms the basis for research. Whether you are in pharmaceutical technology department, pharmacology department, biotechnology department, wherever you are working, the basis is always the chemistry. That's why a lot of emphasis is given to chemistry and chemical sciences in designing the pharmacy curriculum. In fact, not only in India, everywhere the pharmacy has origined from the chemistry only, and later on, because it has now become a very specialized knowledge, now separate faculties, separate departments have come. But together only it leads to success. And then, so today, when Sarvan Kumar has asked me, why can't you participate, sir? I am very happy that Vidyaniketan, the name is very familiar. Your MD, Mohan Babu, is known to everybody. Not met him personally, but I've seen his films, a number of films. Okay, film actor is very much popular. I like him also. And earlier, some 25 years back, not 20, 25 years back, probably when the pharmacy is newly started, I visited that college several times as examiner for undergraduate and PG programs. And later on, with time passes, the examinations are confined to only local people because of many constraints are there, financial matters. And hence, the order of the day is the examiners are confining only to local areas. Whatever it is, I know about this Vidyaniketan. I am very much impressed with the way in which they are used, they conducted the project works because that time continuously for 3 to 5 years, I evaluated the dissertations and excellent work was turned up. And so also is the case in several of the institutions. In the academics, research is going on. But is it all the research that is generated in the institutions, is it effective? Is it fruitful? I can say no, not at all. Because maybe the purpose of our doing research in academics is only to train, particularly to train the students in the basic skills of research, conducting experiments, making interpretations, and writing a scientific writing, and to develop such other skills. The research component will help to create originality, thinking power, and creativity. Such things only the research is very much helpful in academics. But what is needed is the academic research should also reach the level of application, level of industrial research. So industrial research is always application oriented, patent oriented, and in the field of pharmacy or chemistry, almost all patents are from the industry only, because industry always they concentrate on the applied research, application oriented, because that is their business. It is one way, it is their business activity. Unless they generate a novel molecule or a novel delivery system, device, Unless it is patented, they cannot survive in the competitive business world. And that may be the objective. But now in academics also, we should, now it is the time now that we should aim for application oriented research, especially now our attempt should be towards achieving patent. When you will get patent, you know very well that only when it is a new novel, something new concept, or something new element should be there. If it is a routine, nobody will give you patent and no use of that. Everybody know. And hence, patentability should be our aim here. And now in the assessment of the research is done by the institutions, by the individuals at all levels. In the assessment, now the number of patents earned by an individual or institution are counted rather than simply the number of publications. At one time, it is good number of publications. So we are slowly transforming, and for this, every college, big or small, should engage in doing. We do have the facilities. I cannot say no facility, but facilities are there. But how best we can use it properly, and the managements, the people on the top, the CEOs and the principals, they should always encourage the individual teachers and students to do good quality research and aim at patentability. Then certainly progress will be there. And remember that now it is, we are all, not only in India, everywhere, globally, people are waiting for a vaccine. Because otherwise treatment for this viral infection, vaccine. So it takes time, but all are looking at India one, and hence India has become number one in drug manufacturing, sales, exports, as well as R&D activities. And hence the entire world is looking at India only. And now you may be knowing in recent times, okay, a vaccine. So industry people are trying to put out, but the universities are also coming up. Universities are also coming up with vaccine, successful vaccine, and of course all these vaccines are in the phase 2 and 3 clinical trials, and it takes some time to come out. And we wish that in the Indian universities, Indian colleges of pharmacy, chemistry, biotechnology, such development will be there, and soon we will also contribute significantly in this aspect. It is not moving. Sarwan Kumar, it's not getting moved here. Okay, sir. Okay, release. Now this is, so anyway, we are discussing about the research and the type of research that is most needed and most useful, and that we are all engaged in the pharmacy circles is the drug development research, drug development process. And it is one characteristic of this is multidisciplinary research. You cannot say only pharmacy graduates or chemistry graduates are eligible. Equally needed the services of variety of subjects. The origin of this is in the chemistry and molecular biology. And of course, in chemistry, several of the pharmacy colleges are strong, but molecular biology virtually were practically nil. Pharmacy college. I'm not commenting on the zoology and biology, biochemistry departments, but in the pharmacy, even today also, we ignored the importance of molecular biology, and hence the requirement, immediate need is that we have to strengthen our knowledge in the molecular biology along with other pharmaceutical sciences. And that is the origin, what is happening at the cell. Okay. And knowing that only, it is possible to design a molecule that fit into the receptor at the cell and bring a change. That change, the positive way, is the therapeutic change. So like this, it is all including the knowledge of chemistry and molecular biology, biochemistry, biotechnology. So earlier it is only the chemical based drugs only. thousands of new drug molecules have come up. And during the last 15-20 years, a change has taken place. That is a different type of drugs called the biologicals. These are large molecular protein and peptide drugs, which are entirely different in their chemical behavior as well as when taken into the system, how they interact with the cell, and it is all different. So different technologies are needed right from the development of these biological products exemplified by vaccines and sera. A number of antibiotics belong to this category, enzymes, medicinal enzymes, and all these are found to be now the life-saving drugs, but they are very critical. And they are immunogenic also, immunogenic. They bring about interactions, unwanted effects during therapy, and hence it's not enough to evaluate them during the developmental process, but after that also we should take care. Somebody has to follow vigilant over the drug, what is happening, what is it doing in the body, and that is otherwise pharmacovigilance. So pharmacovigilance has become a very important science today, and this is all because of the development of many biological drugs, which affect the immunity system, alter and bring about several reactions. And so this is important. And then the other formulation development and the process development, manufacturing. So engineering component is also very much needed to develop a drug. So drug development means it is just not enough to design a new molecule, but it should be evaluated thoroughly, and it should be industrial also, whether it is possible, process has to be developed, controls have to be developed. All these form an essential component in drug development. And of course, the new chemical entity developed should be characterized, and hence now uh, utmost significance is given to the physical characterization, like crystal form, crystal habit, and size even. Size, all these things are very important. Now polymorphism, all these characters are to be evaluated thoroughly, and now it is known that they are also significantly influencing the therapeutic efficacy of a drug. Simple parameters like the solubility, partition coefficient, in fact, they make a lot of difference. And hence, in the drug design, the chemical modification, after developing a new chemical entity, several times it is not pursuing the adequate quality, pharmacokinetic quality it is called. Then the solubility has to be changed, the lipid nature, lipophilic nature has to be changed. If the result, the partition coefficient, based upon the partition coefficient only, a suitable molecule is generated. All these, the principles of chemistry are to be applied in developing the drug appropriately. And of course, before going to humans, there is a lot of experimentation in animals. So animal pharmacological evaluation, more important is toxicology. And hence the concept is that it is not enough the drug is effective, but it should be safe. And hence safety is given utmost to it is importance in all these evaluations at every stage, initially in the animals, later on in the humans in a controlled way in the clinical studies. So one is safety or toxicity, and next is the therapeutic efficacy, and of course the compound should chemically be stable and integral. Otherwise, if it is degraded or undergone any change, it is of no use. Instead, it may be harmful also. Like that, these characters are to be thoroughly evaluated. This is all the evaluations done are known as a preclinical part of evaluation, and clinical ultimately in the humans or under the supervision of a competent physician or a doctor. The groups will work out, and in a phased manner initially the phase 1 with the limited subjects. Phase 0 is also there. Initial standardization trials to identify the dose trials, etc. in a limited number, 10 to 15 only. Then phase 1 emphasis is made on toxic observations and therapeutic observations, useful observations. Then this is also limited to a particular group, maximum 30 humans. And with related to the vaccine, now the world is concentrating. This phase 1 is completed on several vaccine preparations, developed, and phase 2, that is enlarged. The size, population size is enlarged to 300 in different areas, etc. In phase 3, the same concept, the toxicity and safety and interactions, and observed over a large number of people, patients like ranging to 3,000 to 5,000, not in one area, one particular area, but at different areas, in different countries, in different lands, because to evaluate whether it is suitable for use in the human race at all times. And hence, like this, the research data shall support the safety and efficacy that is essential, and the research data obtained should be reliable. That's very important, reliable data. And hence, research has to be done in the appropriate way, in the scientific way. Guidelines are to be followed. So ICH has given several guidelines. GLP, good laboratory practices, has given another guidelines. For clinical trials, good clinical practices, and in the drug regulatories, okay, the Drugs and Cosmetics Act prescribed such guidelines, not only for the manufacturing, also but for the R&D development activities, and especially quality control. And hence the objective of this entire process, which is a multidisciplinary research, is to develop safe and effective drugs and drug products. And is it possible that a single person can do? Certainly a single person cannot do. A single institution cannot, because it is very costly. It is very costly. Huge amounts of time, enormously long periods of time are needed, and hence only a multinational company come out as a sponsor, and the work is delegated to various other groups which are competent to do all this type of activity. And a recent trend in the last 15 years, 20 years, so CROs, several contract research organizations has come in India. Okay, many are there in Chennai, Bangalore, Hyderabad, Ahmedabad, etc. So they are not manufacturing houses; they are only completely research organization, and they employ the experts only of all disciplines, and they carry out. So the sponsor can delegate, so outsource a part of the work. You develop this up to this intermediate, and then various groups they will give, and every group will work, and finally the sponsor will evaluate which is beneficial to them, efficient and cost effective. Cost, at most underlying factor is the cost always. And like this, so some CRO will be engaged in developing analytical method. Particularly today, at most weightage is given for bioavailability, bioequivalence, biological testing. So you need a more sophisticated analytical equipment and methodologies, and that should be properly developed for bioanalysis. Is another important area that is very much needed for drug development. A third, bioanalytical, LCMS. It's not possible to estimate now the drug and metabolites in the biological fluids. So many advances have taken place in all the related areas, and now is it that in every college and every faculty should equip very well in all the areas? Certainly not, because in the B.Pharm, MSc or M.Pharm programs, the overall knowledge, training, skill is given. But depending upon his interest and his opportunity, and he has to specialize in one area or other, and try to equip more intensive knowledge and latest skills, and certainly we can contribute a lot to this. And then important, so among all, there is a stage somewhere in the middle. After developing a new chemical entity, now I should try to formulate a delivery system or a dosage form, because we see now even the common man know about only the tablets, injectables, and vaccine, a preparation, a liquid preparation, or a liquid preparation given parenterally. That's only we see in the market, and doctor, patient, everybody familiar with them. But it contains inside the active. That pharmacy graduates, chemistry graduates know that it contains inside a powerful drug, and hence the drug developer should be converted into a suitable delivery system. That job is otherwise known as the formulation. and it is an integral part in the process. And hence, this formulation, the pharmaceutical companies consider it as a very important fact, very important. The importance is with the help of formulation only, the company can achieve patentability. So patentability normally, so the chemical entity is one only, one or other inventor company may take a patent and later on others manufacture and market it. That product they will purchase others. Because in India, for example, we are very poor, weak in the bulk drug development and manufacturing. We are still depend upon bulk drugs, China only. China is number one. So we still depend on China only. That's the problem now, to whether to accept Chinese products or not, another issue. But still the dependency is on China. Our Indian manufacturers get the bulk drug and try to utilize it variety of formulations. And because why they more in India, not only in India, globally also, many pharmaceutical companies, they more concentrate on formulation development rather than bulk drug development. Bulk drug development involves an extensive research is needed to develop, but this formulation research is not that much expensive and resourceful, but it is the advantage is always patentability, easily you can achieve. Even though the drug is old, if the formulation is something, some character it is different, you make it more effective, more bioavailable, more usable, some practical convenience of usage. Anything is attributed, and then you can get a patentable exclusive right to market it. And similarly, the life cycle of the drug is elongated. So in one form it is used, later on you can continue it in a different way. You can formulate it into a different system and use it, and hence the life of the drug is in a way elongated. And success of the product depends upon the formulation. Not inside drug is, of course, it is there. It produces the activity. But by formulation, we can also enhance the therapeutic efficacy. At the same time, we can reduce the unwanted effects and hence make it more effective by formulation means. And hence the formulation is considered a new drug product and patent will be issued, and all the companies are doing in that way. So for industrial pharmacists, the formulation development, all the industry people, they consider the very much interesting to them. Of course, for formulation research also you need all the knowledge and services, just as we discussed earlier. And hence, what is this formulation? What type of activities come under exactly the formulation or formulation development? One is pre-formulation. Pre-formulation, and pre-formulation means that all the properties are to be evaluated thoroughly. Unless you know the physical, chemical, pharmacokinetic properties of the drug, bulk drug, okay, you cannot further make use of the drug further, either to formulate it or to put it to use also, it's not. And hence a lot of effort is given today to pre-formulation, in which and analytical method development. It is all well known. Analysis on the basis, okay. In the pre-formulation itself, a method development is taken up first, because for any further evaluation, we need a quantifying method. And hence, the quantifying method should be first worked out. Then, using that method only, you can further study the other characters like solubility, dissolution, bioavailability. To study all this, we need basically first a quantification method, that is analytical. So, analytical method development, and that method developed should always be evaluated, validation, and in R&D activities, whatever you plan to do, it should be validated thoroughly. The equipment are to be validated, procedures are to be standardized and validated, and SOP has to be developed, and they should be used in the same as per the SOP. Everybody should use the same thing. Then only the result obtained is reliable and reproducible. These are the characters very important in research: reliability and the reproducibility should be there, and hence the Similarly, the characterization, NC, new chemical entity characterization of all the properties. Excipients are certainly needed in the preparing the dosage form, and hence the excipient screening, which excipient is good, which everything that should be done appropriately, then dosage form down, depending upon the various considerations, we'll see a bit later. And then assessing the delivery options. If you have a few options are first worked out, and then assess them and select the best. Okay. And similarly, assessing the options and the delivery devices or systems, and solving the formulation problems and issues that become, because all drugs are all chemical materials only. Excipients are also chemicals. Though excipients are only selection is based upon the inertness, inert materials only selected. Even then, it is possible that sometimes some manner they will be affecting, and hence screening has to be done between the compatibility and such problems. They should be assessed, and the process development and development of quality control methods is also research is needed, and in process quality control and quality assurance methodologies are to be worked out. All these activities are done in the formulation department of a pharma company. And similarly, develop this is QC, etc., and packaging development. It's a now it has enormously developed, because packaging, that is the essential purpose, is to protect the drug inside. Okay, because drug is exposed, the preparation is exposed to various environmental conditions, various rigors in transportation, etc., and hence to withstand, to protect the drug, packaging is essentially needed. But now packaging also, there are different ways and means of making attractive and to improve the elegance and to make it more attractive. Okay, first appearance also counts much. That is called technically pharmaceutical elegance. The product should be pharmaceutically elegant, first of all. Then, of course, should have all other qualities, but this is an added quality. So packaging designs, differently type of packaging designs for making the ease of administration, for ease of transportation, for ease of the carrying by the patient, etc. All those things, the packaging development also is now a technical, a well-developed area of science in the pharmacy itself. And then stability testing. Of course, the drug, one of the important character of a medicinal product is that first is that it should be effective, it should be safe. The third one is the quality. All the three are equally important. It is of good quality, and hence the drug should be stable. If it is undergoing any decomposition or anything, it's not suitable. So this stability testing forms another important aspect. And then you have, so is it always that much simple? No, there are several challenges in formulation and product development process. The first challenge is regard to stability. Now within bracket you find certain percentages are given, like 40, suppose the first one is 49%, next one 43. What is that means? It is the result of a study, a survey conducted over 500 pharma companies engaged actively in the formulation development, and the organizers is a part of the WHO program, and they developed various questionnaire, and then their interest is what difficulties the industry are facing related to the formulation and the product development. Okay, and hence the data is analyzed, and these percentage opinion, like it is given in terms of percentage. So 49% of the participating companies, their units, they said stability is the major challenge. And of course, as a science pharmacy graduate, you will also know very well that stability, we are seeing last slide also we have seen, it should be given utmost importance. So stability issues count much in the formulation development, that you should be able to resolve it appropriately. Next is the low bioavailability or poor pharmacokinetic profile. This is another important, low bioavailability. So bioavailability, it is low because of various reasons, and hence how to increase it. Unless it is bioavailable sufficiently, you will not get the therapeutic effect. And similarly, the pharmacokinetic profile. The pharmacokinetic profile means the blood picture and ADME characters, are they adequate to support the efficacy and to avoid the toxicity, to reduce toxicity? Is it this kinetic profile adequate? So now many times the pharmacokinetic profile is not good for the newly developed chemical entities, then certainly the formulation scientist has to do something with this. Next, cost. Ultimately, everywhere the cost is considered as a basic concept, a basic cost effective. And generally, because of excipient, the product cost may not increase. The low cost materials only, low cost alternative, that is the aim always. Of course, drug cost, it is definitely will be there, but the added cost is due to the excipients and other additions and processing. So these things are to be minimized to keep the product cost effective. And hence they identified nearly 40%. And then the next is the drug product safety. Safety is a safety, the drug developed in that, it may be the safe only, but during other manipulations done, ultimately several times the safety may be altered, and hence this is an issue in the formulation development unless you take care. Such issue, depending upon, there is no generally each one is individual, each is different in its chemical character, in its physical character, and hence the issues will also be specific only. Specific. And similarly, low aqueous solubility, similar to low bioavailability, shelf life, it is similar to stability in a way, and patent protection. This is also a problem because aim of a pharmaceutical company is always to get a patent. Why patent means marketability, to get more exclusive market. And then similarly viscosity, excipient compatibility. Okay, all these like these are all the challenges. Many of them are the technical. Technical means using the scientific knowledge. The pharmacist is in a way, the formulation pharmacist can resolve these challenges and make the formulation come out successfully with a good formulation. And global competition is there, and low productivity due to extreme clinical testing. So clinical testing, okay, it appears for normal person that why it needs so much clinical testing enormously, the time is delayed. Now what is happening today is people are eagerly waiting for vaccine, and the manufacturers and government regulating body is telling that not yet over, it takes time. It is true, but for a common man, why so much time? So much time is they are taking, etc. And hence, what is needed is these are the challenges, technical, and they should be resolved by the formulation pharmacist using their knowledge, skill appropriately, and hence, like this, we have a lot of challenges here. And then the opportunities. Yes, this formulation is really an encouraging area. So Indian pharma sector offers a wide range of opportunities, well developed in this area, and hence in the pharma companies, in the CROs, and several manufacturing units, CROs are established throughout the country and offering good opportunities for younger pharmacy graduates to do affairs, as the students are also involved in the symposium. The students, okay, they are having excellent opportunities in this area, and hence they should they could develop the suitable knowledge and skills appropriately to seek employment and prosperous in this area. Like the types of research, what type of research activities that are very much needed? The purpose is that a similar type of research activities we can plan in our academic institutions. These are the normally in Indian pharma sector. these are the activities that are going on in the formulation wing. And what are they is generic drug product development. Generic drug product development that is most widely followed because India is now the number one in generic drug manufacturers. It is supplying the generic drugs, exporting to all countries, advanced countries, generics only. And there are, along with India, the another country where this more largely done is China. Now, the China, because of the COVID-19, so as a bad remark now, the people are afraid of anything taking from China, either science, technology, whatever it is, or our product, anything. And hence now there is no other alternative except India for the entire globe, world. And we are number one in the generic formulation. That is particularly for ANDA, abbreviated new drug applications. And for generic product, the manufacturer need not engage in the entire research. And majorly the type of research is only formulation research because it is aimed at a particular product, a type of product. And hence for marketing authorization, the ANDA. So it can be quickly completed. It's not that much costly and can easily be done, but at the same time enhances the business potential of the company. So almost all companies are trying in the generic drug product development, and they are very successful. And then the other type is largely what type of products? Because when you say generic product, there are many different types of products are there, and largely the conventional only. Conventional in this sense, the regular formulations of tablets, capsules, uncoated, coated, or fast disintegrating tablets, floating tablets, dispersible tablets, coated pellets, pencils, all these are okay, are the different types of formulations that are aimed in the generic drug development. And hence, now what is needed is, first of all, let us be thorough. Instead of worrying, of course, certainly you should also know about the most advanced type of dosage forms, but their business potential is not yet established, still in the investigatory stage only. But these things are, technologies are well developed, and still there is a scope to develop newer technologies for these type of products and formulations, and hence certainly will be successful if we take up the research activities in this and in certain cases also, for example, coated pellets and pencils. In an academic institution, you do find a lot of problem because many places we don't have the coating pans of a small size. Only the industrial sizes are available. That we cannot engage in academic institutions. And hence, in such cases, we should have a collaboration. We should have a collaboration with some sort of industry, a meaningful collaboration, or with support from industry only. We should establish such a lab in the college. We should convince them. We should impress them that our faculty and principal and institution has all the adequate facilities and expertise. We can deliver the sponsored research appropriately. Then only the companies will come forward. So one thing minimum required for pre-requirement for sponsored research is that we have to prove our competency. We have to prove our competency, expertise, skill, and dedication. And what the people, outside people want is time specific. If they sanction a project and earmark, say 1 year, in 1 year exactly you should complete. You should complete. It's not academic. Academic means if the project is not complete, the universities are good enough to give extension. UGC and CS are also good enough to extend the fellowship, tenure of fellowship. But in industry, it is not like that. If they prescribe a time means within that time, we should be able to complete. So we should develop that skill that we should stick to times. So this is industrial collaboration is certainly needed for certain types of formulations when they are something above the normal level. Like your pencils, coated pellets, etc. Other things like conventional tablets, etc. You can easily do anything. anywhere. Only thing is that the size, enlarging the size. Then again, you need the support from industry. Then liquids like syrups, including suspensions and dry mixers, is one area where still number of products are coming up dry because of the stability characteristics. Many are made into dry form, suitable, converted into liquid form just before usage, and hence they are using. They are again using. So similarly, modified systems, injectables. Among the injectables also, it is the lyophilized products. You have to concentrate on lyophilized. So you should need a basic requirement is a good lyophilizer. Good lyophilizer. Without a lyophilizer, you cannot think about these lyophilizer systems. All the modern drugs in the sense that enzymes, proteins and peptides, biologicals, all these biologicals are lyophilized only, and hence and all our nanoparticulate systems are also lyophilized only, and hence for developing such type of products, again, a minimum basic requirement is lyophilization, very well utilized, and lyophilized products also offer advantage biopharmaceutically, means that the dissolution absorption will be more rapid, and hence additional advantage will also be there. So lyophilized products are increasing for parenteral use particularly, and similarly modified drug delivery systems. These are various types, in the same manner: sustained release, control release, extended release, as quoted matrix tablets, etc. There are spansules. All these are also very much well used for generic drug product development. And many times the products made are only coating, by coating process only, and hence the coating technologies, though it's difficult in an institution, you should aim at, you have to modify, you have to change our philosophy of thinking, and impress the management to develop such labs which are at least equipped with pilot scale type of scales, and engage in the coating. Coating is an essential process in industry for all purposes. It's very rare that today you see any tablet formulation without a coat. Coating is there for. Okay, I'm happy that tomorrow my student only, who is that TEGK Murthy, from Bapatla, principal of Bapatla College of Pharmacy. So I'll see in the topic is very, very interesting. Coatings for enhancing the dissolution rate. Normally coating, we simply think that coatings are to stabilize and to control release, to reduce the release, means control release, etc. But he has titled it as it can also be used, and he is having a pilot scale lab in his college. Maybe that more equipped, well versed with that, and I am happy that tomorrow I will certainly join the meeting and follow what he is telling. So this is like this. Now here you have the modified drug delivery systems. Under that, the fancier type of coating, referring to coating. Coating is very useful. So from the tomorrow's lecture title, coating can be used to enhance the dissolution bioavailability. At the same time, majorly coating is used for protection and obtaining control release. And hence, how many colleges are having the facility to apply the coatings and do some sort of work in the coating? Even we are not organizing several 90% or even more 95% college, we could not do the coating for the classwork, and hence where the scope do the research in that area. But a time is now come that we should enhance the capability of institutions and engage, and we should certainly be able to do that type of research, very much useful. And then NDDS, a lot is read and projected. Okay, and industry is also giving weightage importance, that is novel drug delivery systems. But they are still in the developing and research stage only. You can do certainly, I'm not saying that you should not do. You should do equip. But industry people, they are very big, like bulk drug, like the basic drug development research, it is big companies only they are manipulating, and they are doing in the same manner novel drug delivery. We studied a lot about liposomes. Okay, every B.Pharm student and M.Pharm student will read in pharmaceutics liposomes. Of course, nanoparticles has reached the commercial success, commercial stage. But nanoparticles coupled with not available as particles themselves and as suspensions, but lyophilized systems. Certainly available. But other things, though a wonderful research is being done in different different areas of different NDDS systems, other than now really novel new, commercially they are not that much coming to picture. The reason is that still in the developmental stage only. Commercial means practicality, feasibility should be there, and cost should be effective, and hence certain concepts which is studied under this novel delivery systems, it's all not feasible and practicable and cost effective, and hence still we have to wait some more time. But certainly you can, for enhancing your quality of your work, etc. Certainly you can do research in this area. Similarly, in the other areas include process development, including quality control specifications and methodologies. Somebody has to do workout. Somebody has to work out process development methodology. Okay. Now the dissolution rate. It appears to be simple test now. The pharmacopoeias, but anybody is concerned with the if the dissolution rate of a highly potent drug, the dose itself is 2 mg. It is contained in a tablet. Tablets can be made, okay, potent, highly potent. But when you study the dissolution, what is the method of assay you can follow? Is it UV method is not suitable? Then HPLC also not suitable. So if that is the dose is such a low quantity, just as in the plasma analysis, here also, even in the dissolution also, now you find problem. Then how to develop, or what other methods can be applicable, and develop a method and validate. Validation is utmost important whenever there is a development. And hence, the analytical method development for assay, dissolution rate, stability studies, and their validation. Stability studies also, not all the methods are suitable for stability study. Okay, only the method which in the presence of degraded product should selectively estimate the undegraded product. Okay, or the degraded products impurities should be identified. So for all such purposes, the highly methods are still needed to be developed. So it's an analytical profiles are needed to be developed. Again, analytical impurity profiles. All these are the areas which still a lot of research is needed, and certainly the academicians also can involve, provided we develop the necessary facilities. And stability studies, bioavailability and bioequivalence, of course, it's a clinical study, cannot be done in any institution or even in industry also, only in a hospital should be done. But certainly we should equip ourselves for conducting these, understanding the principle and doing in the animal, okay, preclinical studies should be very thorough. And then in vitro, in vivo correlation. This is an area which is very much needed regulatory wise, and lot of work can be done again in this area. This is all again done with the formulation department only, and dissolution, because in vivo performance is only on one product. That they will do in the bioavailability or bioequivalence study in a clinic, under clinical setup. But to develop a matching in vitro method, which gives a good correlation with the in vivo data, we do need a lot of experimentation to alter the dissolution, okay, methodology, and hence evaluate, and ultimate evaluation is made which dissolution test procedure is suitable to make such decision. The data is correlated with in vivo, and based upon the which method gives good correlation, that those conditions are identified and fixed as the method. So like that, there is still a lot of scope for doing the in vitro dissolution method development with the ultimate aim of achieving in vitro in vivo correlation, etc. So these are the areas of research that is taken up in the formulation development. So formulation development does not simply mean that preparing a tablet or a capsule. Okay, all these areas are involved research. Actually, the formulation pharmacist can deliver things positively in all these areas. And then, so this is okay. Next one. Next one is now we'll see after knowing some concepts of formulation development, etc. Now we'll see the formulations are generally you can identify it into two categories only. Two broad, that is conventional dosage forms, and another is controlled release, including novel or whatever you call. Okay, all the two categories you can distinguish. And first we'll take up the conventional. So conventional, briefly, the essential principles used in developing those dosage forms is so this is all for conventional formulation development. The concepts are based upon the biopharmaceutical classification system, BCS system. So this is essentially BCS system. All the drugs are divided into four categories based upon two essential characters. One is solubility and the other is the permeability. So these are the two important physical properties that influence the ADME of the drug and hence the therapeutic efficacy of the drug. Okay, and they should be balanced. And hence, based upon these two characters, the drugs are divided into class 1, 2, 3, 4 like that. Okay, and class 1 means both are high only, and hence virtually they do not pose any problem. There, even whether you make a tablet or a capsule or a solution, both will performance will be the same when administered, and hence virtually don't have any problem with regard to them. Next, second category means low solubility, high permeability. Low solubility, high permeability. So here the problem is with regard to low solubility. Permeability, inherent lipophilicity is okay. The molecule can easily cross the barrier. But low solubility, this is limited. Because of low solubility, the first step required is that it should be soluble. So here the student should remember, unless the drug is soluble in water, unless the molecules are available in solution at the absorbing surface, the particles cannot cross, only molecules can cross. That is the basic concept. Particles cannot, whatever is the size, particle cannot cross the barrier, and it should be dissolved, and in solution, molecules are said to be available. The molecules can cross the barrier provided they are lipophilic enough. And hence the dissolution or solubility form the rate limiting step for several molecules, and almost all organic drugs, by virtue of their organic character, all organic drugs, synthetic, okay, they belong to this. They come under this category 2 only, which is class 2 drug, and hence dissolution is the rate limiting step. So that is the reason why today the dissolution can given a very significant role in biopharmaceutics, in the formulation development, as well as in understanding the therapeutic efficacy, etc. So class 3, just class 3 means it is permeability is low, means inherent permeability character is low, whereas class 3 and solubility is high. So here again the problem is not with solubility but permeability. So permeability you have to make use of that, and what dosage forms are needed for such drugs. And similarly in the class 4 both are low, and hence they create a number of problems. And hence what options you can make in such cases, that is very much needed for a formulation pharmacist. And hence you are talking about highly soluble and low soluble, etc. So what do you mean by highly soluble? Means the highest dose soluble in less than 250 ml of water at the different pHs that you come across in GI fluid. So in the GI tract, you have the pHs ranging from 1 to 7.5 at different regions. And hence at all these pHs, the solubility you have to evaluate, and basing on the data, if that obeys the highest dose, okay, soluble in less than 250 ml, and hence we call it as highly soluble. So they do not create any problem. Otherwise, but surprisingly, you have acidic as well as alkaline pHs in the track, and hence one compound, particular organic compounds classified as weakly acidic, weakly basic. One is soluble, one may be soluble at acidic pH, but insoluble in alkaline pH. The one which is soluble in alkaline is insoluble in the acidic pH, and hence for almost all drugs, solubility will become a problem when given orally. So that is the reason why the solubility has formed an important concept, and you have to necessarily tackle that issue. And to see that sufficient solubility is exercised and good absorption occurs. Because absorption is the first step to derive the therapeutic efficacy. Then similarly, high permeability. Permeability is the character of how well it is crossing the biological barrier. And we know that lipid molecules cross the barriers rapidly because the biological barriers are all lipid in nature, whereas surrounding fluids are all aqueous in character. And hence, in the drug design itself, this is considered, the concept is considered lipophilic nature. So lipophilicity has to be introduced into the molecule if it is not sufficiently lipophilic. Because several of the drugs, they absorb majorly by passive diffusion, and hence the lipid solubility should be adequate. And lipid solubility at the same time, we have seen earlier, active solubility also important. With the result, a balance in the sense partition coefficient. That is the reason why the partition coefficient, okay, O by W, the two immiscible partitioning between two immiscible liquids can be assessed outside in the lab. That forms a very important parameter in drug design to check the validity of the molecule developed, whether it is suitable or not. An ideal partitioning is between 0 to 3 only, 0 to 3. That means defined as K O by W. That means 0 means equal miscibility in water and lipid that rapidly absorb, very rapidly. And that one is like an example cited everywhere is alcohol. Alcohol, so when you administer alcohol, it rapidly gets. But now this permeability is so important character, and how to assess. It's not only the permeability just not equal to partition coefficient. In addition to partition coefficient, lipid solubility is one important factor. Aqueous solubility also counts. At the same time, molecular size is also important. Molecular size also important. And of course, organic drug molecular size are low, suitable for easy penetration. But coming to the modern biological drugs, their size is large, and hence that is the reason they do not have the permeability character. At the same time, they are rapidly degrade because they are unstable, mostly unstable, and hence the problems will come with such drugs. Today delivery systems are being developed even for the administration of such drugs orally. So how to identify whether it is highly permeable or not? It is a case related, case wise you have to evaluate, and hence this is taken. The basis for this is F. F is a pharmacokinetic parameter, fraction absorbable, and that fraction absorbable, okay, is calculated by comparing the availability of oral route with the IV route. And if it is greater than 0.9 or 90% absorption, then it is considered as satisfactory, highly permeable. If it is less than that, then problems will occur, and hence basing upon this criteria, the classification is done. Next is these are the same characters are written, okay, for all the 4 categories, okay. And the first category, okay, the bioequivalent studies are unnecessary. That is one important consideration. Because if you prove that it belongs to category 1, means that you have to assess the solubility character mainly at different pHs, because you are telling higher soluble, highly soluble not only at one pH. The problem is at all pHs, And hence, if that is the case, it belongs to class 1. And in such a case, there is no need to perform any bioavailability and bioequivalence studies. Okay, approval will be given. And so it is all behave in the same manner, and they are more suitable for control release. And the rate limiting factor in the absorption is so not the dissolution now. It is not the dissolution, not permeability, etc. But it is a gastric emptying. A third biological factor of importance is because the barrier between stomach and intestine is only passes, only emptying, how it is rapidly. Okay, that forms the rate limiting step. Okay, and here similarly in the class 2 means again limited dissolution rate, active solubility is limited, and here dissolution. So you need enhancement of these characters, solubility and dissolution rate, and they are also suitable many times because several organic compounds are in this category only, and well suited for good absorption given orally and suitable for all types of formulations. And in the class 3, and class 3 means permeability is low, so you need so modern way develop several lipid based drug delivery systems have been developed. They tackle this issue. The lipid based formulations enhance the permeability of the drug, and hence such things like self emulsifying drug delivery systems, micro emulsifying, nano emulsifying systems, all those are very much suitable for class 3 drugs. Class 4 drugs. Class 4 drugs is unpredictable. You cannot predict because both are low. Both are low. And hence, as far as possible, then alternative routes are selected, alternative ways of administering the drug, because unpredictability should be avoided. Okay, if it is low, it should always be low. It should high, it should always be high. But in between is unprediction means the performance is highly variable, and hence such things are not at all accepted in the practice. So like this, you have the this, okay, and this is again application of this principle. This BCS classification is very much useful for delivery system development, okay, and this is of course we discussed the same thing. Next, okay, now this is class 2. Many 95 to 98% of the modern drugs are today organic only in character, and hence they need some treatment. Okay, processing. Like one is micronization, and today it is nano sizing. Okay, nano size. So the simple fact is that in both the cases, size is enormously reduced. When size is reduced, normally the dissolution absorption will be very fast, and hence micronized drugs are made available not now. It started, this principle is identified as early as in 50s, and molecules are made available, compounds are made available in micronized form in starting from 60s itself. And hence, so many formulations, so many manufacturers use the micronized form. So traditionally there are different techniques, but still milling is the only technique feasible. Different types of sophisticated mills are available for all scales, large scale, medium scale, etc. Laboratory scale also they're available now, and hence we can conveniently convert the drugs into micronized form, evaluate the particle size, and if you make use of them, normally the performance will be very high. Why very high means the absorption character enormously increases. That is the reason. And when absorption increases and high blood levels are reached within a short time, that will help us to reduce the dose also. And with several drugs, it is demonstrated that, okay, by reducing the particle size, okay, you can greatly reduce the dose required to achieve the same level is also well established. Similarly, the nano sizing. So nano size still this small, but the stability problems will come. Provided the stability problems are avoided, today it's easily avoided by using lyophilization. Okay, no coalescence again, dried in the same form, and can be converted into tablets and capsules, and they are very much useful because in addition to size reduction advantage, the nano sized particles can readily cross the barrier. They can readily cross the biological barrier. Okay, that is a age-old principle. No, not new. It's very old, older than all of us. Okay, that is we studied colloids in chemistry. in physical pharmacology. Colloids are nothing but nano. That time the word nano particle is not there, only colloidal, less than microns. Colloids. Colloids are found to exhibit wonderful penetration and found to be very good antimicrobial. Okay, colloidal gold, colloidal silver, all those things are well known, established. The reason also attributed, the particle penetrate well into the organism. That means biological membrane. So same concept in the modern way, nanoparticles. So the two reasons put. One is the particle size reduction, increased surface area, and then greater resolution absorption. Another is the particle itself can cross the barrier, whereas micronized particle they cannot cross. They should undergo dissolution. As molecules only, they can cross the biological barrier. So the other technique that is normally followed is adding a surfactant, addition of surfactants. So that is certainly surfactants are added in small amounts in all the tablets, capsules intended for oral use and other purposes. Okay. So these small quantities, they enhance greatly the wettability of the compound. Because organic compounds, again, we are discussing about class 2 means they are lipophilic, means they are hydrophobic, and hence they do not easily wetted with water. So when administered, all fluids are aqueous in character, and hence the first step itself is not happening. Wettability is important. So in such a situation, addition of a small surfactant will tend to improve wettability. That means by reducing the interfacial tension, they increase the wettability. So when wettability is increased normally, the dissolution is enhanced, rate of dissolution. So addition of small surfactants. So many surfactants are developed in recent years. Among them also, non-ionics are more used because non-ionics are they are non-toxic. Non-ionic surfactants like Soluplus, okay, Poloxamers, Gelusair, all those non-ionics, they are like trade names, of course, and non-ionic surfactants are more used commercially and because they are non-toxic. So we can also easily get the samples of pure compounds and use it. Then the other is formulation as emulsions and microemulsion systems, that is as self-emulsifying systems. So they are wonderful formulations, particularly for class 2 as well as for class 3 also. They do well. A number of investigations proved that, and still they are also available in the form of commercial products, and hence this is all applicable for class 3 also. Use of complexing agents, like the most successful complexing agent commercially is cyclodextrin, beta cyclodextrin. Today you find several manufacturers using beta cyclodextrin, and a lot of research is generated in this area, cyclodextrins. And earlier, though it is thousands of research publications you will find, patents you find in this area, cyclodextrin complexation, because it is suitable for enhancing all the characters of the drug, solubility, dissolution, stability, whatnot, every character is improved. Because it has a beautiful molecule, forms inclusion complexes. But it's not used till 2000. The reason is that it is very costly. Because earlier, producing it by chemical means is very costly affair. And in 90s, in mid 90s, etc., a fermentation process or a biotechnology means, microbial conversion means is developed to prepare these cyclodextrins. With the result, the cost of the product is enormously reduced, and hence after that, around 2000 starting. So almost all companies, they use cyclodextrin in their formulations to enhance the all characters, particularly bioavailability problem. All insoluble drugs like simple drugs like pyroxicum, okay, it is clubbed with this and marketed. Even some they use it to write CD on the label also, though they are not supposed to write it. Okay, excipient name we need not write on the label. Only active ingredient name is enough. So it is so complexing agents is another process, and hence For generic formulation development, the options available to you is the best way is mechanization. Mechanization is milling. Industrially milling is possible, but pharmaceutically you have the other technique also, like preparing a solid dispersion, solvent deposition. That also particle size greatly reduced, but stability comes into it. But these solid dispersions can also be made industrially using the other techniques. Other techniques in the sense, okay, by making so use of like one is lyophilizer. Okay, lyophilization is always a versatile technique. Wherever the stability problem is there, conversion problem is there, you can immediately convert into that. So the and hence the complexing agents also do well. This is class 3. Yes. So peptide and proteins constitute the part of this class, and the technologies handling such materials are in the rise now, because many more drugs are coming up under this category, and the lipid-based systems are being developed for this, and or otherwise you have to alternative is only that parental route, and this is the same thing with you have to go for a change in the route of administration when both are low, and hence this is a list of all the methods and technologies for control. Because that BCS classification system works well with the conventional type of dosage forms, okay, that are under the generic drug formulation category. And this is the other way, other type of products are known as the control release. So you know very well what are these. So there are different systems used widely, like diffusion control systems, like matrix tablets, microcapsules, microspheres, etc. Dissolution, again, preparing poorly soluble complexes, resinates, pellets, etc. Microcapsules again. And release controlled by ion exchange, resinates, resonate complexes, they are also useful. And then osmotic pressure controlled. They are very successful commercially also. Oros, osmotic pumps, and infusion devices, etc. for oral as well as for parenteral. And hence this is the control release technologies many times they based upon coating or otherwise matrix embodiment. Okay, and each one has its own advantage, disadvantage, but to improve the elegancy and to get the reproducible performance in the in vivo system, the coated pellets or spancel type is mostly used. That is the reason why in the market you find many such things. But coating, especially spancel manufacturing, is rather difficult. A number of parameters are to be controlled, and but whereas matrix is a simple technique. Matrix compression is a simple technique, but serves the purpose. The only disadvantage labeled with the matrix system is that after administrating, it exhibits all or none effect. Okay, because it's a non-disintegrating entity, and then it may stay either in the stomach or emptied into the intestine. Suppose depending upon the drug PK and other values, okay. Suppose if a drug is more absorbable from intestine, small intestine, several drugs do like that only. And if such a drug in a coated pellet is tablet is staying only in the stomach, though it is released slowly, you will not get sufficient absorption, and hence it is ineffective. So to avoid such problems, gastric emptying problems, okay, the spancels are coating at particulate levels, like spancels somewhat big, and coating at particle level like the microcapsules and microspheres. They are done. That they are beautifully empty gradually, and hence their performance is more reproducible. So these are the and otherwise they go for using the osmotic principle, and continuously the drug is released through the narrow orifice made on the coating, and different designs are available. You can have a chance to do good amount of work in this area also. And then like this, oral products, you have the matrix tablets, same thing, coated pellets, swallowable matrix, different types of matrix are also possible today. Each one has its own advantage and disadvantage, and oral osmotic pumps and mucoadhesive tablets, etc. And in the label, you may not find the word mucoadhesive. Nowhere tablet is labeled as mucoadhesive. But it depends on the excipient used. And if a large proportion of a mucoadhesive polymer like HPMC is most widely used polymer for several purposes, and it also adds to this character, and carbopol, it also adds this character mucoadhesion. And in the track, it adheres and increases the duration of stay of drug, dosage form and drug availability. Mucoadhesion and adhesive dosage forms are now designed for almost several routes, and is always advantage. The main essential advantage is only that they prolong the release, made available over a longer period, because especially when oral route. Otherwise, the dosage form unit may be emptied in 8 hours or 10 hours, and during that time the drug may not be sufficiently released, reducing the total bioavailability. In all those cases, to prolong the residence time of the system in the GI tract, mucoadhesion is the best principle used. And then the microcapsules, coated pellets, have been successfully used for controlled release products. So in the market, whatever controlled release product is available, it is all prepared by these techniques only, either sustained release or controlled release, etc. And hence, you have the novel drug delivery systems also. The word novel drug delivery. Under this, you have the systems like liposomal drug delivery systems, sustained release systems, drug delivery systems, and nanoparticles, nanocrystals, iontophoretic devices, and then pumps, osmotic pumps, implants, all these implantable devices, all these are considered as novel, new and novel, in the sense that they are serving the different purposes, either sometimes enhancing the solubility, bioavailability, or making the product more stable, or really extending the duration of effect, etc. But essentially, a new concept, novel concept, when they use it, it is called a new polymer or a new type of entity is employed. It is considered as a novel drug delivery system. And essential purpose is to reduce the toxicity and to enhance the efficacy by targeting mainly, and hence such specialized systems all come under this category. And a good amount of research is being done, but commercially not many products are now available, only highly specialized products, like for example transdermal drug delivery. You see, so much work has been done in the literature on transdermal drug delivery, patches and other things. But commercially, no one, not even a single product. At one time they introduced and they withdrawn because of commercial feasibility is not there, cost is not effective, and such reasons are also considered apart from the technical issues. Technical mention. Briefly we'll discuss, and hence the other characters, those who are interested in generating an equivalent product to that, should first analyze the innovative product. That is first step. Then selection of the route and type of product. That's very important. Route and type of the product, and developing the standards or specifications. This is another important. So before starting formulation and then manufacturing, etc., so you have to develop the standards. First step is what standards do we need in this product? The standards in terms of the physical, chemical properties, and pharmacokinetic profiles, as well as the therapeutic quality. Suppose if your aim is to get relief from the pain or a headache, so consider a simple case of designing your product to give relief from headache. Then what is therapeutic need is immediately should relieve the pain after administration. In 5 to 10 minutes, you should get the relief. And hence what it indicate? A fast absorption, fast achieving the concentration, and immediate onset of action should be very short. That type of formulation. So for that, you need an IR tablet, not a sustained release, IR, immediate release. So in the earlier one of the slides, we have taken what are the techniques for immediate release. So such techniques, so for example, in the initial stage, microfined aspirin is the first example. So the term itself indicates micronization. So you will get a very quick relief from the headache, and it is very successful during the last 40-50 years. It is still in the market, and hence like this, okay, the what quality you need in the therapeutic quality. Similarly, pharmaceutical quality you need. How much, okay, disintegration time should be there? How much dissolution rate? That is today for all 99% drugs formulations, dissolution is critical. For both soluble, insoluble drugs, it is critical, and hence you should think about what dissolution you need to fix. Okay, you have to fix. For that, normally the innovator, the researchers will copy the innovator product. That is the reason why the innovator product analysis is done. So innovator product is giving 80% dissolution in 20 minutes. So that they put it as the target and target. Okay, like that. Okay, so specification, disintegration, it should be like this. All qualities you have to fix up the standards. Okay, physical, chemical, particle size. What particle size is more appropriate? You have to fix up, and all standards you have to establish first. Then you should try to build up the quality in the product to achieve all these standards in your product. That is formulation development. And then the selection of methods and techniques. Sometimes you need some technique. Without that, it is not possible. So anyway, what techniques? Even not specialized techniques like complexation, you can also try to do. Even the tablet making also, you can have the different techniques of granulation. Okay, at granulation, dry granulation, direct compression, using directly compressible vehicles, different types, etc. So you have to select some technology used. Do you need a surfactant or not? Like that, all things using your expertise, knowledge, and require taking into consideration the physical, chemical characters of the drug, etc. You should work out developing the specific selection of the methods, techniques, and more important is excipients. What type of excipients you need, etc. And selection of excipients. If you need a super disintegrant, yes, there are many super disintegrants. Which one you select? There should be some rational basis. Today, otherwise, it will not be accepted. Regulating bodies are so rigid now. So for every inclusion, you should give some reason, rationality, and hence, if you say I am using 2% tall, you should give one reason why you are using 2%. Okay. And selection of excipients and their levels, and developing form. This all leads to developing the formula. Based upon the formula, again, process development. Identify the process which is most suited to manipulate this and to get into the final form. And then processing of trial batches certainly needed, and evaluation of the product to conform to quality. Trial batches you have to perform, and then enlarging the formula, and at each and every stage you have to confirm whether all the qualities identified as standards specifications are there or not. And hence these are all the different steps that are involved with more or less whatever is the type of the dosage form. This is the sequence of understanding needed. And then formulation development and concept of quality. Here comes the quality, concept of quality. Where today quality is considered utmost important. It does not mean earlier it is not there, but today it is surfaced. The reason is because of globalization. So you heard the word global. Because of globalization and open marketing, competition, highly competitive business, and hence everybody think about quality only. So what is quality is simply conforming to all the specifications. All the specifications are laid down by you only. And hence you check up whether those specifications are obeyed are there in the formula or not. So when the product conforms to all the specifications laid down earlier and got approved by the regulating body, okay, and later on when you produce the drug, it should conform to those specifications only, then the product is said to be of good quality. And then these regulating bodies are there everywhere. Everyone want to check the quality. And quality of what? Not only the finished product quality, but the quality of the operations, process that you are using, whether it is of good quality or not, as expected. So regulations are there. That's why the concepts of quality assurance, pharmaceutical quality assurance have surfaced now. So globally, different countries have their regulating bodies and they made rules and regulations. Okay, and GMP guidelines are given, laboratory guidelines, chemical, for everything guidelines are there. Validation take, for validation also it is not as we like we can do. So guidelines are there, ICH guidelines are, WHO guidelines are there. So you have to conform to these guidelines and perform. So all those ensure that quality is there in the product. It's not only simply final finished testing of finished product, whether the process, how it is done, etc. That's why today there is a good requirement of obeying the raw material testing. Raw material testing you have to do. Documentation is needed for every thing that is done. Okay, at all stages, validations are to be done. Okay, process equipment, even personal validation should be there, and SOP are to be developed and followed exactly, and evidence should be created that whether the person is following that particular SOP or not, and automation has come for that purpose, etc. Routinely, all things are to be validated again and again, and in-process controls are to be used, recorded everywhere. So all this is all the modern regulation. They are to ensure quality only, and hence quality, that type of quality is at most important. And then, so the first thing is that quality cannot be added like an ingredient. If sweetness is there, you can add some sweetness, artificial sweetener or a natural sweetener. But quality cannot be. So quality has to be inbuilt into the product at every stage, right from the quality origin is in the formulation development. And another point of origin is in the manufacturing facility. How to create the manufacturing, how to establish manufacturing facility. Everything guidelines are given now, and accordingly only how to do. So important. Suppose earlier, when say myself a student or a principal sir is a student, okay, we have not studied about the in detail about aspirin tablet manufacturing. We read, but only the thing is that those days everywhere it is a dry method has to be used because aspirin is very sensitive, very sensitive. A dry method has to be used, otherwise it will get degraded, etc. Okay, dry granulation method is. But later on over years, now the student is well familiar with the concept that relative humidity has to be controlled. Now the essential rule is that aspirin products are now to be made at a relative humidity less than 30%. Less than 30%. That is the condition prescribed globally. Otherwise, it is not accepted. And the manufacturer can say, okay, sir, we are following. Nobody will accept unless evidence is created. Document should be there. So for to get 30% humidity, what is needed? Now, a day like this today, anywhere in the country, the humidity will be around 75 to 80%. That humidity has to be reduced to 70%. Okay, a dehumidifier is needed, otherwise it's not possible. So dehumidifier, okay, what technology is used, etc. It is left to the manufacturer. That, but he has to introduce the concept of that. Okay, dehumidifier into the manufacturing area where aspirin tablets or products are supposed to be made, and then record the humidity and keep a record of the humidity daily. Then when inspection comes, they have to prove that, then only that facility is accepted. That means quality also depends upon the facility needed. Facility should be created more appropriately. Yes, that is the reason why earlier small manufacturing units used to be plenty in India. Now, after the introduction of GMP mandatory in 2004, 31st December, that is the deadline given. After that, it is made, before that it is only advisory. Okay, now it is mandatory from that time, 31st December 2004. Okay, so later on, compulsory, all these things are to be followed, guidelines prescribed. Earlier it is not mandatory. I mean, those who are interested will follow, otherwise not needed. But now global competition is there, and hence because of global competition, okay, if not our government, because the manufacturer want to supply it to US, UK, Australia, so those governments will send their inspectors and approve the product. So then only these manufacturers can supply, export the drugs to other countries. That is the reason why. So the concept of quality, though the concept is there since long, but it is now taken utmost care and has surfaced, and the quality has to be built into the formulation apart from other things. Apart from other things means infrastructure, of course, assuming that good infrastructure needed is there as per regulation. Okay, good expertise is there as per regulation. Okay, and hence comes as a formulation pharmacist, you have to think about quality right from the first stage of developing the formula. Otherwise, it is not possible. Suppose if you want to generate a simple tablet having a DT, disintegration time, is it less than 1 minute. Now the student is well known today that all the commercial tablets, uncoated tablets, they disintegrate within 1 minute. Okay, several times within seconds only. So that you have to make it as a specification or a list of specifications initially, and basing upon that list. So to be quality, your tablet should disintegrate within 1 minute. So for that, you have to think about what material I have to add in the formulation. And in the formulation, necessarily should include a powerful disintegrant like a super disintegrant in appropriate concentration. Otherwise, it is not possible. If you simply use a starch, dry starch, any form, it's not that much adequate, suitable. Say super disintegrant, you have to make use. Or a material like a bicarbonate or a citric acid combination you have to make use of, effervescent combination you have to make use of, in which case immediately the tablet will burst. So that means the origin is in the quality. Unless you think this concept while formulation, you cannot achieve the specification of DT less than 1 minute. In the same manner, if your specification is to achieve 80% dissolution in 15 minutes, the drug is a highly insoluble drug. Okay. There are so many drugs are there. They will never give that dissolution. If you prepare a tablet, regular tablet, then you have to think about either any technique that we have seen earlier, a complexing agent or a micronization, etc. And certainly you can manipulate other things. And selection of every ingredient will affect the dissolution. The binder and its concentration, disintegrant and its concentration, even lubricant. Okay, lubricants are hydrophobic. They are very dangerous enemies, dead enemies to dissolution, because they are lipophilic, talc, magnesium stearate, all are hydrophobic, and hence carefully the selection is made, thinking the quality in mind, and hence quality, the origin of quality is in the formulation only, appropriately, or to develop the formula. And levels also to be fixed appropriately. For that, what is the way? What is the way? And now comes formulations to achieve the desired quality. Desired quality. Okay. Design or formula shall be on a rational basis. That term we are using frequently. Okay. It involves selection of excipients and their levels majorly, and techniques also to achieve the desired quality. And this is normally achieved in formulation development by two means. One is the traditional methods. Traditional methods means trial and error. Trial and error in the sense that suppose a tablet, the critical component is binder, another critical component is disintegrant apart from the drug properties. Okay, and hence you evaluate, make an alternative, a series of formulations. Okay, altering the binder, evaluate them and select the best one. Then similarly, other ingredients also you go on making. So initially, number of trials are made, varying one or other variable or one of another variable at different levels, and testing them, and then select the best addition. Okay, that is the traditional. the trial and error method. But in this technique, it is a large experimentation has to be done. A lot of experiments have to be done. Laborious, time consuming, wasting resources. At the same time, you will get only the individual effects. Individual effects in the sense, the effect of disintegrant only you will get, or effect of binder only you will get, or lubricant only you will get. But when in a tablet, when they are present, combinedly they produce effect. The binder and disintegrant, combined effect. Binder and lubricant together they may produce an effect. That is known as interaction effect. Just in a chemistry laboratory, when a reaction is influenced by, say, two factors like temperature and pH, it is well known that the temperature and pH, they are having their individual effects. To evaluate that, okay, temperature is evaluated 4 different levels, pH is evaluated for another 4 different levels, and then select the best pH, best temperature, etc. That is traditional way there. But though the experiments are done in that way, a number of experiments, but still we don't know what is the combined effect of pH and temperature. Together they produce an interaction effect always. And hence in the same manner, to know the interaction effects, whether it is in a chemistry laboratory or in a formulation laboratory, the best design of experiment is the factorial design. The best design of experiment is the factorial design. So factorial designs are there, and which are very much helpful for the purpose of evaluating the individual effects as well as the combined effects of 2 or more factors. And hence the use of modern approaches like QBD and optimization. So in both the QBD and optimization, the same DOE, okay, experimental designs, design of experiment. DOE means design of experiment. That is, instead of studying individually, so selected combinations of the factors involved are identified, and they are known as the factorial experiments. With the help of factorial experiments, you can easily find out the individual effects as well as the combined effects. And hence, basing upon the data, that okay, so basic requirement of QBD. QBD is quality by design. So design should ensure quality in the product. That is a much widely used word in industry and regulatory affairs. QBD approach. It is simply QBD approach is nothing but design experiments using the DOE principles, majorly the factorial experiments. Because wherever there are 2 or more factors, it is possible to expect a combined effect, not only the individual effect. And hence, the simple experimentation is not at all suitable, DOE. And the experiments are designed as for factorial experiments like 2 factors, 2 levels, 2 factors, 3 levels, 3 factors, 2 levels, many combinations are there. And then when the data accumulated is very large, when variation. So for each factor, we have to carefully select the range of variation. You have to select the range of variation. This range of variation is critical. That is, otherwise, in technical sense, it is called QBD, critical space. That means the range should be selected carefully to evaluate its effects. And hence, in this range, 2 levels of range, the experiments are done. Similarly, for each factor, you have to space, okay, 2 levels, giving the space, and hence a combination, selected combination of these is identified by factorial design and implemented. Okay, and in the case of QBD, the results are analyzed by multiple regression analysis, and the effect of the dependent variable. Dependent variable means your dissolution rate or your yield of a chemical reaction. It depends on 2 or more independent variables. Independent variable means temperature and pH. Here, the concentration of one excipient like binder, another excipient like a cyclodextrin, etc. Okay, so this is the worked out as per factorial design. Results are obtained and then they are analyzed by multiple regression technique, and different forms of graphical representations are generated, which will give an understanding of the effect of individual effects as well as combined effect of the factors. And from that, different combinations which are needed to achieve the desired response to desired effect are identified. Okay? That is QBD. And the same thing is otherwise known as optimization, which are almost the same meaning. Optimization. Optimization is the word used. Okay, optimize. To optimize means, okay, to achieve what is desired. To achieve what is desired, that is not to achieve maximum. Maximum is not needed. You desire something, you achieve, try to achieve that only. That is optimization. And hence these techniques are today much popular and most needed. Why these designs are used today is to satisfy the regulatory requirement, because regulators now ask evidence for every inclusion. If you say that I am using this much level of cyclodextrin, they ask you what is the reason, why. If you use more, what happens? If you use less, what happens? Where is the evidence that it gives the best result? So that evidence is created by DOE, and hence the optimization and QBD more or all same. The inherent principle is using the factorial experimental designs, analyzing the data by multiple regression technique. Regression analysis, we are familiar, that is only a relationship between X and Y. Linear regression means the relationship between X and Y. Two variables only, one is the concentration and another is optical density in the colorimetry or spectrophotometry. In the same manner, multiple regression means two variables will be, two independent variables, and it's both will influence okay one variable, and that is known as a dependent variable. And so there will be two or more x's and there is only one y. And what sort of relation, mathematical relation exists between these two is developed using the multiple regression analysis. Okay, and hence the basic principle is there. So now I titled it as optimization methods, and most widely used optimization and simple optimization technique. It is also known as QBD approach only. Both are ideally same. I explained the difference. Okay, and hence here I already discussed to achieve the maximum, etc. So in a factorial design, because instead of going through every aspect here, I will essence only I will tell now. So in the factorial design, okay, this is either they are designed as a 2 to the power of 2 or 2 to the power 3 to the power of 2. Means 2 factors and 2 levels is most widely used. There are 2 factors, many times 2 factors, and to evaluate the effect, that factor has to be studied at minimum 2 levels. That is enough. So 2 to the power of 2. And hence when it is a 2 to the power of 2 experiment, okay, now you need to expand 2 to the power of 2. If you expand 2 to the power of 2, it is 4, and hence 4 treatments you need. The 4 selected combinations of the 2 factors. In the same manner, there are 3 factors. There are 3 factors, 3 to the power of 2, each again 2 levels. So 3 factors into 2 levels. Again, it is expand that 2 to the power of 3, it is equal to 8. 8, and hence 8 selected combinations of the treatments you have to develop using the different levels of the 2 factors involved. And hence the one example given here is formulation of IR tablets. IR means immediate release tablets, and this is beta CD, starch 1500, and Solu plus. SP means Solu plus, are considered as the 3 factors for optimization of IR tablets of according to 2 to the power of 3 factorial design. Okay, in each case. The 3 factors and the 2 levels. Now how to fix up the levels? So in the table, you find the levels. So factor is, first factor A is beta CD, second factor is starch 1500, third factor is Solu plus, level, lower level. Lower level is indicated by the symbol -1, and higher level is indicated by the symbol 1. So you have to carefully select the levels, okay, for assessment of their effects and combined effects, and later on for optimization. And hence here it is, the levels are for beta CD. Generally beta CD is used as its usage strength is given as in molar ratio. Okay, that means 1:1 ratio, 1:1 ratio of drug and CD. Molar ratio of drug is to CD. Okay, on that basis, so the lower level is fixed at 1:1 and the higher level is fixed at 1:6. That means for every one molecule of beta CD, so drug both are equal. Okay, and in the case of 1:6, that is drug is to one for every one molecule of drug, 6 molecules of beta CD are fixed. So you may ask, how, why you have to fix, how you have to fix this. This is after when you have a knowledge about this, and after reading more of the informational literature on the cyclodextrins and evaluate in which proportions they are normally useful. So basing upon that, if the proportion is even less than 1:1, okay, it is not adequate, because here the concept is should form an inclusion complex. And hence, after a thorough survey of literature, evaluate in which proportion they are normally used. Out of that, depending upon the practicality of your case, etc., dose and other things, you fix up a suitable range, but not too narrow or not too wide. An optimum range like this, 1.1 and 1.6 or even 1.5 also will do. Okay, but suppose if you fix up 1.10, 1:10, now the bulk will be enormously large, which is not practicable for compression. And hence, like all other considerations, also you have to take into account and fix the dose levels. Then the other thing is it is only a starch 1500 a powder, a directly compressible vehicle powder, which is hydrophilic, and this is 2% and 30%. Because it's a diluent normally, directly compressible vehicle diluent, and hence we can go even up to 30% otherwise. Okay, and 2 to 30%, 2 on the lower side, higher side 30. And then SP, soluplus. Soluplus is a surfactant, non-ionic surfactant. The purpose is to enhance the dissolution, etc. And it is 0 and 2% are fixed. Normally, a little surfactant quantity will be very, very low. If you add more also, the problem is technically in the making tablets compression process, the tablet will be too soft and hence it is not accepted. So, but a small quantity is enough to serve the purpose of increasing the contributing to enhance the dissolution. So it is 0%. 0% means we are not adding any, and then 2% means 2% of the total in the sense drug plus CD. Basically, the proportions also have to define clearly. Like this is otherwise in the QBD, the same thing is known as fixing the space only. So these are the things, and hence it is the design is a factorial design, and it is called what type of factorial design? 2 to the power of 3. And when you expand 2 to the power of 3, it is equal to 8, and hence 8 combinations you need. So they are known as 8 IR tablet formulations. In each case are formulated with selected combinations of the levels of the 3 factors, and that is how to write down. That is like this. So here it is, okay, and this is the tablet is as a to explain you the I selected this work. Tell me certain tablet. So this is an insoluble drug, VCS class 2 drug, and for critical dissolution, and hence dose is 40 mg. All tablets contain 40 mg only, and the ingredients are: one is beta cyclodextrin, next starch 1500, soluplus, and other things regularly used. They should keep constant like talc, magnesium stearate, total weight, etc. Okay, total weight. And now this is the total weight here cannot be kept constant. You can try to keep it, but proportions will alter, because additive proportions are based upon the percentages, and hence when you change the excipient, that to beta cyclodextrin, the range is wide, 1:1 to 1:6. Okay, and hence quantity will be large, and and hence it is not possible to get the same weight for low dose drugs. Okay, still low dose drugs you can maintain that. So now it is F1A, they are designated as formulation FA, F1, 1A, B. How to remember and how to generate it quickly, whatever is the case, There is a uniform pattern in statistics provided. So first start with 1, write 1. Then write A, then B, then product of A and B, AB. Then introduce C, then AC, BC, ABC. This is a sequence you can remember for 3 factors, ABC. So the figure that is there in the notation indicate that factor is at the second level. If there is no figure in the notation, that indicate that factor is at the first level. So in the symbol 1, there is no A, no B, no C. A, B, C are missing, and hence all the 3 factors are at the lower level only. So now we check up what are the lower levels. So beta cycloreduction lower level is 1:1, means 1 drug 1, 40, 47. The second one is starch 1500. So what is the percentage selected in the earlier slide for starch 1500? 2%. And hence here 2%. 2% of what? 2% of the total mass of drug and beta CD. Because beta CD also takes a large proportion, and hence together the total bulk. Out of that, the starch 1500 2%. And hence 80 or 2% of 80 is 1.6. And then, okay, then soluplus, it is 0. 0 means you need not add anything here. And hence in the next A means A factor is at second level. A means at beta CD. So beta CD at second level means 1:6. And hence, what is 1:6 times 40? That is 240. Okay, that figure is 240. And then the A factor at second level. The remaining are at the first level. Remaining means B and C are at first level. And what is B? B starts 1500, first level only, 2% only. And what is 2%? 2% of 240 plus 40 means 280. So 282% is otherwise equal to 5.6. 5.6. Like that percentage you have to work out. Okay. And then in the first series up to AB, there is no C. And hence dash. No surfactant added. And then suppose in the same manner, for example, for C, this is FC, formulation FC means C is at the second level, A and B are at the first level. And hence A means beta CD, first level is 40 only. And the next B also at first level, starts 1500, that is again 1.6, because it is 2% of 40 plus 40, 80. And then soluplus, that is at second level. So second level means soluplus, what is second level? 2%. And hence again it is 2% of what? Drug and beta CD. And hence it is also 1 point. Like this, you have to work out all the formula, all 8 formulas. Okay, 8. Okay, up to ABC. The last one is optimized form, that will come at later stage. So this is the way in which you have to work out the factorial experiments into formulation based after fixing the spaces for each, and these are the alternative formulation. So how many we are doing? We are doing only 8 formulations. Okay, in the same procedures you have to follow, and then after that, when we evaluate, these are the dissolution curves for the because to find out the differences. Just as separately given first 4, 1 A, B, like that is this one 4 set, and this is another 4 set, and same using that the parameters. So parameters means, okay, these are the 8 F1 to FAB, like that up to FABC, different formulations in which the CD is varying, all the 3 factors are varying, and they're not randomly, it is on a rational basis as per the design DOE. So parameters of importance is PD10, that is 1, and T50, DE are all routine, other things are routine. Anything you can do. But for the criteria, for a specification has to be laid down initially. And the specification for tablet formulations, especially when the dissolution is critical in all these cases, tablets, compressed tablet. Okay, dissolution is the best parameter apart from other things. Other things may be taken as the normal standards, but dissolution is critical. And accordingly, you can select. Or you yourself can judge and select an appropriate one. So here the selection is that the fixed is 85% is taken as the required target PD10. PD10 means percent dissolved in 10 minutes. I fixed it as 85%. This is big, 85%. This is not compared anyway, innovator, etc. That is not the idea. So innovate, we fixed it. Why it is means, as such, telmisartan is a BCS class 2 drug. And when a formulation of any drug is above 85% in 10 minutes, it is considered as rapidly dissolving and biowaiver can be given. That is the regulatory rule. Okay. And hence, in such cases, after verifying those rules, now the 85% is considered as the target dissolution to achieve in 10 minutes. That is specification. Okay. Now, this is based upon this PD10. So, these are the, for all 8 products, there are 8 PD10 values are there. And now, basing upon this PD10 selected prefixed standard, you have to go on doing the optimization. Optimization means you have to identify, okay, which of the combination of the 3 will yield the required dissolution, like 85% dissolution in 10 minutes. That is what is needed. And otherwise, to achieve the optimized dissolution or optimization. And here for that, the technique employed is analysis of the data by multiple regression techniques. It is a simple, it is a statistical technique. It is similar to your multiple regression analysis, but many times we do only in the classwork only X and Y, only linear regression only, Y is equal to mx plus c type. But here it is, there are, how many X's are there? 3 X's are there, X1, X2, X3. Okay, like means each X is defined already. One is beta CD, another like that. And so 3 X's and 1 Y value only. What is Y response? Y response is the response means the PD10. We are taking it as the required parameter for optimization. And hence PD, that is Y, the third column Y, response Y. Okay. That is percent dissolved for each formulation. For 8 formulations, it is given. It is given. So, for example, first formulation 35%, dissolved in 10 minutes. Next 43, next 96, next 89, 37, 42, 95, 82, etc. So, some may be more, some may be low, because variation is there in the formulas. There's so much variation is there, and hence whatever it is, okay. And sometimes it may be nearer. Even then also, using the relationship has to be developed. Relationship means multiple regression, means you have to develop a mathematical equation for the variable Y, response Y, Y is equal to in terms of the X1, X2, X3. Okay, that type of equation you have to develop. For that, the technique is multiple regression. This multiple regression, so this is done in this way, and the transformed potency. Transformed potency means instead of using the actual potency, we are using the lower potency is always designated as -1. In the earlier table also I have shown. In the higher potency, higher level is indicated by the positive +1. And hence here it is for the first formula, for example, formula 1. Formula 1 means what are the levels? All the 3 are at lower level. And hence I have written -1, -1, -1 in all the 3 areas. Okay. And formula A means A is at the second level. And hence A, under A +1. Whereas the other 2 -1, -1. Like that, you fill these columns. It is nothing but whatever you have decided earlier as levels, first level, second level. Instead of using the level as 1:1, 1:6, you use it as -1, +1. Because this is universal. Whatever is the problem, it is applicable. Later on, those minus plus ones will be ultimately converted into the real potencies. That is what is done. So then it is Y is written corresponding Y. For formulation 1, Y is 35. For formulation A, 43, like that. For formulation AB, it is 82. Okay. And then the sequence of operations, they are known as the response multiples, are to be worked out. That means the A means x1, B means x2, C means x3, and hence here multiply each x with the corresponding y. And hence on the top what you get is the label x1y, x2y, x1, x2y, x3y, like that, okay, and the above. And in the same manner, minus 1, first thing, minus 1 into 35, minus 35. So all minus 1, all, so next is x2y, that is also minus 35. x1, x2y, how it is 35? x1 multiplied by x2 is minus 1 multiplied by minus 1 plus 1 into y is 35, so plus 35. Carefully you have to do, carefully you have to do like this, okay, all you have to work out. And then each column like x1, y, it is one multiple. Next multiple is x2y, x1, x2y, like that all. Ultimately, last one you will find x1 multiplied by x2 multiplied by x3y, all the three products. Then okay, now carefully, because some in each, now in each column, some will be positive, some will be negative. Magnitude is the same, 35 always. Okay, some will be positive, some will be negative. And hence, carefully sum up, these are the sigmas. Sigma total divided by number of replication. Because here each factor is studied in 8 experiments. Total 8 you have, 8 sets of experiments you have, and hence R is equal to 8. So divide sigma divided by R is equal to, those are the values. Okay, average response. That is first one is minus 0.875, next is 25.68, like that. Okay, you will get. These are known as the coefficients. Because in a mathematical equation, before the variable, you have a coefficient, and hence this technique, first the coefficients are found out, and hence first beta coefficients are known as the beta 1. Beta 1 is the coefficient for the x1 term, okay, and beta 2 is the coefficient for x2 term, and third column beta 1 2, beta 1 2 is the coefficient for the term x1 into x2, like that all these are coefficients, okay. And hence the type of now the, okay, we will go to next slide, okay. Now that is the equation is always y in terms of x1. So what is it we have estimated as beta 1, beta 2, beta 1 2, or they are the coefficients, and hence you substitute the coefficients there, y is equal to, so x1, starting from x1 you have already coefficients from the earlier table. But how to determine the 64.44? It is called a x0. It is called x beta 0. Beta 0. So for that, okay, after writing all other things, keeping beta 0 as such, and select any one combination. Select any one combination. Among the 8, you can select any one combination and substitute that y, okay, and hence rearrange it to estimate the beta 0. Beta 0 is the coefficient. Okay, it is not related to an intercept, otherwise known as in the linear regression. Okay, so this is the equation. That is the mathematical equation. What is it we have done? We developed a mathematical equation giving the relationship for the response in terms of the variables, 3 variables, x1, x2, x3. That is the equation. Okay. And I am using the first table, transformed potency. There are some important concepts, that's why. So there, because minus 1 plus 1 we are using, that how it is derived is transformed potency, it is called. This is equivalent to x, real value of x, minus the average of the two levels divided by half of difference in the two levels. That will yield to Sorry, another 5 minutes I will close. In another 5 minutes. 5 minutes. Okay. Okay, that means, so these are transformed potencies. So we are using all the analysis using the transformed potency -1 +1. How you arrive at that is using this equation, transformed potency. So x minus average of the two levels, okay. And first beta CD two levels are 1 and 6 in a way, 1:1, 1:6. So 1 and 6. So average of the two levels, 1 and 3.5. And by half of the difference. So again take the difference and divide, it will be equal to first level is like that. And for example, it is given for A. So here it is A, not exactly in the earlier case, but suppose a factor is there 2 and 30. And then a lower level is low. How to find out the transformed potential of lower level is given here. Lower level is equal to 2 minus 16. What is 16? 16 is the average of the 2 and 30, 32 by 2, 60. Okay, by different half the difference. The difference of the two levels. What is the difference of the two levels? 2 and 30 means 28. 28 by half means 2 means it is 14. So the lower level transformed potency is equal to -1. 2 minus 16 by 14 minus. Similarly, higher level of the same factor is 30 minus 16. 30 is the higher level, actual level. And minus 16 means the average. Divided by 14 means half of the difference. Again, it is +1. Like this. So whatever is the level, you have to use this formula to convert the real potency into the transformed potency. At the end, again, if the transformed potency is known, you should be able to convert into the actual potency. Again, rearrange the same thing. And hence this equation is very much useful. And then to determine the optimum levels, how to determine the optimum levels, that is important. That means first fix the levels of one or two factors depending upon the number. So you can fix one or two, anything. Two, first you fix randomly as you like, within that space only, not above, below. Within that space only means within the range initially fixed. Within that range, you select one for one factor or two factors, and then using the mathematical equation, you estimate what is the magnitude of the other factor left to achieve the desired resolution. Desired response is 85%. So why should be 85%? And suppose in the next slide, we'll see the same thing. So take x1 as 0 and x2 as 0. That means transformed potency 0. What does it mean? Transformed potency 0 does not equal to numerical 0. It means the central value of the two levels. Central value of the two levels. That's all. Okay. And hence x1 factor, okay, that is CD. Two levels are 1:1 and 1:5. The central value of which is equal to okay 0, which is equal to 0. Central value. Central value is okay. Central value of the two is equal to 0, and hence in the same manner, okay, for x1 is taken as 0, and also now for the other things also you can do in the same manner. For example, the soluplus is fixed at 1%. Soluplus is fixed at 1%. Means to estimate x2, substitute 0 and the equation reduced to so first thing is that this is the equation. In the earlier slide, it is there. Wait a while. So the equation developed is y is equal to 64.888, etc. That equation. Now you fix any two things as 0. Transformed potency as 0. 0 means not numerical 0, it is central value. So central value 0. Suppose x1 is taken as 0 and x3 is taken as 0. Means in the above equation, wherever you find x1, put 0. Wherever you find x3, put 0, substitute, and then solve it. Then two factors will become 0. Wherever you find x1 or x3 will become 0. That means then the equation reduces to y is equal to 64.88, then x1 0, delete that. Next x2 is not deleted, x2 is you have to estimate the optimized quantity, you have to estimate. So 25.62 x2 be there like that only. Then another time x1 x2 that will become 0. Again x3 is assumed to be 0, and hence it will be, and it will also go. And similarly other things, wherever you find x1 x3, that term will become 0, and the remaining equation will contain only x2. And then you solve that x2. What is that x2? That you have to do. And that is what is done here. And then finally the x2 is okay. This y, what y will take? Y is the specification needed. That specification needed for this is 85% resolution, 10 minutes, and hence 85 will remain like that. And the other equation reduces to 85 is equal to 64.88 plus 25.62 x2. That rearrange and solve it, and x2 is equal to what is the value of x2 is equivalent to 0.785. This 0.785, what is that? It is the transformed potential, because in the developing the equation we have used only transformed potentials, and hence this 0.785, that is x2 value. X2 refers to the factor starts 1500, okay, B factor, and the transformed potential desired, optimized transformed potential is equal to 0.785. And now convert it into real potential. How to convert? Again, that's why I have given that equation. Again, use the same equation used for transformed potential. For relationship between transformed and the real potential, take that equation. And here it is transformed potential, you take it as 0.785. And with the result, the required, so potential is given, and you rearrange that, and real potential of the factor B starts 1500 is found to be 26.99%. 26. So that is x2 means that is optimized. But what about x1 and x2? They are already fixed at level, 0 level, and hence the same thing is written here. So optimized formula to achieve 85% dissolution is in 10 minutes. That is required dissolution is CD is 1:3 ratio of drug and beta CD, because we assumed 0. Transformed potency is 0, and hence real potency is midway, halfway. It is not equal to 0, numerical 0, halfway. Halfway means the original potencies are lower is 1:1, higher is 1:5, and hence the average of these is 1:3. That is what is written. Okay. And similarly, starch 1500, just now we estimated it as 26.99% of drug is beta CD, and Solu plus also we are taken 1%. 1% of the drug. 1% how it come? How it come? It is also assumed to be the 0. Transformed potency 0. Transformed potency 0 in the case of factor 3. Solu plus is the levels are 0 and 2%. Lower level is 0 and higher level is 2%. The average of that, the central value is 2 by 1, 1%. 2 by 2, 1. Okay, and hence 1%. 1% of what? Drug and CD complex. Because right from the beginning we are defining the percentage in terms of drug and CD, because CD itself occupies some mass. And so these are the percentage, and then transform into the formula. Okay, depending upon the dose. So when you say beta CD 1:3, dose of drug is 40, and hence beta CD is 40 into 3, 120. So, and this is already given, estimated in percent only, 26.99. And then Solu plus is 1%. 1% of both drug and. So first you have to estimate what is the drug and beta CD, then estimate the. So this is so optimization. This is the equation, and then finally these are the quantities, and optimizer formula is given. Okay, this is okay. This is all the same thing, and when tested, you should conform to the required specification. It is confirmed, and now we go to formula. In the optimizer formula also given in the beginning itself. So this is dissolution. Okay, and the last column. Okay, IR tablets, F-opt. F-opt means optimized formula. That means drug is 40 and then beta CD 140 and similarly Solu plus 182. Oh, till one more is there, I think. ABC, okay. Like this, the same quantities here it is written. Now we'll see. The dissolution curve. This is the data. FTIR opt. Opt means 85% in 10 minutes. PD10 is 85%, 85.5% plus or minus 1.25. 1.25. Nowadays, dissolution should accompany any estimate should accompany with the standard deviation or standard error. So, like plus or minus that quantity. And the T50 is in minutes, average is 2.9 minutes. And DE20 dissolution efficiency 20% is 75.7. And the same thing, rate, specific rate is also given. Like other things are okay, but essentially the optimization parameter is taken as a PD 10%. And hence, after arriving at the quantities and prepare the same product in the same manner and evaluation, every method should be the same, then you will find that it is normally it confirms. We worked out with several drugs and several types of formulations, and other the formula. Although this is for floating tablets, but because time is not available, okay, and then the same way of analysis is done, okay, with the result. Okay, now so the optimization like this by essentially it is by using DOE experimentation, that is factorial designs, and analysis of the results by multiple regression technique. Is a simple statistical method it is for optimization without the use of complicated software. And the advantage is that with this method, you will arrive at a formula, single formula. But whereas if you use the software, you will arrive at several formulas. Then the pharmacist is again puzzling which formula I have to take. Because several of my industry friends, students only, industry people, their managers they are using this technique, optimization technique by software, same thing. Same principle in software. So they get a number of alternatives. Suppose in each case they may get 20 to 30 options, alternatives. But alternatives, okay, it's okay. But how to select among the alternatives? Again, it's a puzzle. It's a puzzle. But one way is whatever option is, every option if you prepare, you will get the same thing. But when 30 options you give, the puzzle is that which one I have to use. And hence, it is avoided here. Here also we can generate several formulas in the sense that at the end what we did, we first fixed two things, 2x1 and x3 fixed and x2 is evaluated. In the same manner, you fix x3 and x3 and evaluate x1. Like that, various permutations and combinations you can do and develop the various formulas. That is the same thing done in a software, and you will get a large number of formulas, as many as 30. Okay, and hence again the difficulty is that they could not understand, and even the people again may ask, unless they prepare all the 30 and prove that it is same, they will not accept. Instead of that, here it is very selective, depending upon, okay, if you want to change the strengths, you can change and still arrive at optimization. Only thing is that among the three, two things are one. One is enough. One you can take 0, another you can take 1. Transform potency fixed. And matching to those fixed, the third potency you have to estimate using that mathematical equation. And then the transformed potency is ultimately converted into the real potency. That will be worked out. Percentage you can work out. So like this is a statistical technique. Several types of formulations are being worked out, and even selection also can be done by I have not shown you the analysis of variance technique used here to find out the individual and combined effects. That itself is a different story. Individual and combined effects. When once you know that, you can also make a rational selection of the combination. We can make also a rational selection. Suppose a binder and tablet. In a tablet, a binder, disintegrant, lubricant. What combination will be suitable? Again, the experiments can be designed in the same manner, as a factorial experiment, and you have to find out there the ANOVA, annals of variance, or the factorial experiment, which is typically the same principle but slightly different way. You have to analyze the data to know the individual effects and combined effects. Possible combinations are, when there are 3 factors, the possible combinations are 8 combinations. Whether these 8 combinations are significant or not, which one is significant, you find out. Whose effect is maximum, you find out. Then you can take it as a. So, and hence this technique basically is a statistical method, factorial design and multiple regression of data. Statistical experiment itself involve at the end ANOVA. But straight away I have gone to optimization here because the topic is optimization. And then these things can be successfully used for the rational selection of excipients, to evaluate the individual and combined effects of the excipients, and also for optimizing to fixing the means fixing the levels of the variables. And any type of experiment can be designed and optimized, even not only in this this technique is recommended even for analytical work, chemical work, whatever it is. Okay, and hence for the data analysis. So I conclude that okay, only thing is that initially okay, because it's a combined program. Okay, so, uh, topics like this will require more time now. Otherwise, it will not be possible. And moreover, these things like the involving some sort of mathematics, some sort of pharmacokinetics, etc. So the best way is that the direct contact with the student and the teacher, or the learner and the teacher. And best way is, even though like these slides can be used, but individually, step by step, we have to sit in direct contact with the students. And then I encourage always this type of presentation in the class. But I will ask at each stage, I will ask the student, of course, you are all learned teachers, but this is for all. Symposium means for all, students and teachers together. Okay, then they will better understand. Instead of simply by speaking something and looking at the slide, it will not, they may not catch the point. Okay, and hence, anyway, I thank Dr. Shravan Kumar, professor and convener of this symposium. It's an excellent attempt is made today by the team, principal and convener and other faculty members. I congratulate them. A wonderful concept evolved online symposium instead of keeping quiet at home. So and I find several, in fact, I could not find time also to follow all the so many FDPs and symposia being conducted by various colleges daily. Almost one is other are going daily. Why not one? Simultaneously, 2 or 3 are going on. Now I know that at the same time there are 3 others. Others are some, because the students, my students only ask, sir, I am having that also, sir, this also, sir, what to do? Do whatever you want to do it. Nothing wrong. Okay. So thank you, sir, for giving me this opportunity to interact. So thank you all once again.
Thank you, sir. It was an amazing lecture. Hope all of the participants have many takeaway points. Thank you, sir, for your valuable lecture. The session is now open for queries. And I request Mr. M. Sabaris, sir, to handle the Q&A session.
Good afternoon, sir. Welcome to QA session.
Oh, okay.
So question from Dr. Madhavi Lata. What is the importance of fractional bioavailability in controlled drug delivery systems?
Okay, that's good. Excellent question. Fractional bioavailability means F value, fraction absorbed, okay, for oral products. Now, normally in the pharmacokinetics, you studied this. So the rate of release, if only fraction is absorbing, that rate should be increased by the fraction, but it goes to the denominator. That has to be raised. If suppose 50% only available to the system, then you have to increase the rate also by 50%. So in the kinetic equations, you always find this F value in the denominator. In the denominator. Suppose I can tell that what is the K0 in control release? K0 in control release is estimated based upon the elimination rate constant KEL multiplied by the desired concentration into VD. Desired concentration is CSS into VD. CSS into VD is the amount, and that multiplied by KEL is the elimination rate. And whatever rate it is lost, the same rate you have to input. You have to introduce from the dosage form. And this equation is applicable only when F is equal to 1. Means whatever drug is given orally, when everything is absorbing, that will do. What is that? That means K0 is equal to, okay, first thing is CSS into VD into KEL. Three things only. That will enough. But several times the fraction absorbed is not at all 100%. It's less than 100, less than 1, and hence the equation will modify itself. The whole thing divided by F. F is the fraction absorbed. Okay, that means in such a case where F is different from 1, normally less than 1. Okay, in such a case, the K0, that means sustained or controlled desired release rate, is equal to CSS. Always start with CSS. CSS means it is the desired concentration at which the level has to be maintained in the plasma into VD. Together give the amount, desired amount in the body. That multiplied by KEL is the elimination rate. The whole thing divided by F. If F is, say, for example, is given as 0.8, so divided by 0.8. Means if in the denominator, if you introduce a F value as 0.8, then K0 will be increased. Because any increase in the denominator will increase the variable. So K0 will be more. Is it clear? That equation itself you remember. Then, so certainly, when the drug is given by oral or any other route involving the a sort of absorption, certainly you have to divide, you have to include the concept of fraction absorbed. But several drugs, the fraction absorbed, its extent of absorption, otherwise it would be 90 or 95%, in which case it may not make much difference. But suppose if the fraction absorbed is much less, 50%, 60%, then certainly you have to divide and then increase the rate. Then only things will work. This is all kinetic equations of control release formulations. This is discussed fully. Okay, madam. So I think you understand, madam?
Okay, sir.
Hello, Madhavi Lata. Okay. From which college?
Sir.
From which college? Madhavi Lata.
Madhavi Lata.
From which college?
Sir, Swati College of Pharmacy, Nellore, sir.
Okay, good. Yes, it's a good question. Because there
okay.
I put the word F, but not explain because it is something different. It is something different. Okay, now. Then
okay,
good,
sir.
Good question. Anything else?
Next question, sir, from Dr. Ramesh. Explain the procedure involved in patents for challenges in formulation.
Oh, patent to challenges, sir. Challenges and patents. Sir. No, two things are different. Two things are patenting, procedure for patenting or for challenge. Suppose if somebody announced a patent, we can challenge. We can challenge means in the patenting procedure, when an inventor applies for a patent, the patent officers, they first study and send it to an expert and evaluate, get the evaluation. And then if, in principle, if it is okay, then they gazette it, they publish it, and that is called, it is open for challenge. So challenging, anyone can challenge. Anyone, like who is an expert, who can understand the PD, and he can challenge. When they can challenge, when you say that the information given is already known as, is already known, it is on there, like that. If you give references and prove that, It is already there in the open domain. There is no need for any patent. Like that is a challenge. Okay. So patenting last step is challenge. Challenge means patent office will publish. There is a page patent gazette. All the patents are published in the gazette before final acceptance. Okay, after the expert review everything, collecting opinion, etc., and the experts may raise some questions. Those questions will be given to the inventor, and inventor will give explanation. After every process over, finally, when they come to a conclusion, in principle, we can accept it. Then they publish in the gazette, and then it is said open for challenge. Open for challenge means anybody like us can read it, understand, and we suppose we have any doubt. And our claim is that it is not new, it is already known. It is in the open domain. I know this because it is there like that with evidence. If you challenge, when challenge it is there, no, they ask again the inventor to explain. If you could not give proper explanation, the patent will be rejected. Will not. That is what it is. So patenting, a part of that is challenge. Okay.
Okay, sir.
And sometimes the industry people, they file a challenge petition also. Okay, because it is there, the same meaning as what that one. So it is already known, we have done, we have with our technology is there, we already registered. Like that, they can give evidence and challenge any patent. But the inventor should be capable of defending that and prove that it is their own, in which respect it is different, in which way it is new. If they prove it, they will certainly be accepted. So normally in the business, such things will happen. Normally in the business, because it's a competitive world, business people, they do not agree simply whatever the other person says. They go on challenging on one or other ground, and they should be able to reply it appropriately, prove it otherwise. Okay, sir?
Okay, sir. Next question from Sheikh Ahmad. Which type of analysis techniques used for innovators product?
Oh, which type of analysis means? So the word analysis of the innovator product means innovator product has to be analyzed for different qualities. Depending upon the major concern is all characters, right from weight of the tablet, shape of the tablet, color of the tablet, all along with all the qualities, and finally the evaluation is made for dissolution and bioavailability also. That's the picture. So it depends on the drug, and you have to. So normally, if it is a patented product, in the patent literature, the inventor or developer will give the method which they follow. The method which they follow, they write down in their original patent, because it's all patented products only. That's why we cannot use the same term and the same product after the expiry of the patent only. We market as generic product. By that time, we have to do all these sort of developmental work. The first step is analysis of the vendor product, the innovator product. So innovator product in terms of the character important, all are important, but finally, so dissolution and bioavailability. So bioavailability, depending upon the drug, they have to use a method. And because if you say use your own method, and if you say you can test, because they may say that yes, that method is not suitable. This method we use, it is more powerful like that. If they say, in case, but here the question is no challenge here, because though the one who want to develop a generic product, he will market it only after the expiry of the innovator product patent. Before that, nobody is allowed. By that time, they will be ready with all the procedures and developmental stage, approvals, everything. Okay. And the approval will also be given only when they prove that it is equivalent, biologically bioequivalent to the innovator product. So for that, necessarily a clinical study has to be done. That is the only clinical study needed for generic product development, A&D application. Other clinical tests need not be done. Okay. There, of course, depending upon the drug, a suitable method is written, but in the original patent, they use some method that easily you can get it. you can get it, and the same method you can use better. Then there is no doubt. Because sometimes the performance also depends on the method of analysis, even the in vitro or in vivo. In vitro dissolution critical. There are so many methods are there, but an effective method should be used. And many times methods like HPLC method, depending upon the dose, LCMS methods are adequate. They will do well. It is possible. Okay, there is no that this method only you have to use. It is not like that. But when you apply to the A&D application, it is not again when the innovator product is under patent control. We are not getting any marketing authorization. We are not eligible to market. Only after that expires, then only we have to think about this marketing. And hence there is no question of competing with the innovator, but the regulatory bodies should ensure that the appropriate method is used. And for that, the innovator literature also should be verified, and you can use the same one. Or otherwise, a method can be used provided it is properly validated to the satisfaction of the regulating body. Here it is only regulating body important. Because they are the authorities to give approval, A&D approval. Okay.
Okay, sir. Thank you, sir.
Okay,
Thank you for answering the questions.
Thank you, sir.
Thank you, sir.
Thank
Hope
you.
everybody have cleared their doubts. It's the time for virtual felicitation of the eminent speaker by the convener, Dr. Anna Balaji Garu.
Sir, good afternoon once again.
Good afternoon.
It is our good fortune that you have accepted the invitation and given a very great lecture. Like a true teacher, you have given taken a lot of time with your lot of patience, enormous mountain of patience to explain so many things. This is what our teachers, your contemporaries used to do. They used to take a lot of patience with all of us. So, which is now missing in the next generation of teachers. You are a big example and inspiration for all of us, sir. We are very happy and honored to be associated with an eminent personality like you. As a token of appreciation, our love and affection, we are presenting a small e-certificate which will be emailed to you. A hard copy will be posted to you after the college reopens. Thank you very much, sir. We will invite you once again. Once the college reopens, please visit our College of Pharmacy. And if God permits, we can visit Tirumala also.
Sure. Thank you, sir. Thank you all once again. Good luck.
Thank you, sir. After a fabulous session, I call Professor K. Sarvan Kumar, sir, to propose vote of thanks.
Good afternoon, all of you. It's my immense pleasure to say vote of thanks of today's event. It's my sincere and cordial thanks to our beloved chairman, Padma Shri Dr. M. Mohan Babu Garu and CEO Vishnu Manchu Garu to provide all facilities to conduct such kind of events. I would like to thank today's chief guest, Professor K.B. Chandrasekhar Garu, Vice Chancellor of Krishna University, Andhra Pradesh, to accept our invitation from his busy schedule. Professor K.B.C. Chandrasekhar Garu covered all kind of research activities. It's my sincere thanks to today's godfather of pharmacy, today's speaker, Professor K.P.R. Chowdary Garu, advisor of academic and research in KVSR Siddhartha College of Pharmaceutical Sciences, and research advisor in Vikas Institute of Pharmaceutical Sciences, Rajamandri, Andhra Pradesh. So, SAR has covered so many research corner for budding researchers how to develop the formulation for the innovative developments. So I would like to thank our advisor, Professor Venugopal Reddy Garu, and Quality and Development Director, Professor I. Sudarshan Kumar Garu, to their continuous support to conduct this kind of event. I would like to thank our beloved principal, Dr. Anna Balaji Garu, Vice Principal, Professor Mohan Lakshmi Garu, and organizing committee to giving their support to conduct this kind of event. I sincerely thank to all college principals, delegates, and my well-wisher. Thank you all. Namaste.
Thank you. Thank you. Once again.
Thank you, sir. Thank you, sir.
Thank you, ma.
Thank you, sir. With a big thanks, I would like to call it a day. Thank
sir.
you all.
Thank you, madam. Thank you, madam.
Thank you, sir. Thank you, sir. Thank you.
Awesome. Thank you, sir.
Okay, sir. Thank you, sir.
Thank you.